Look around, not ahead. A peak is just a way to walk at the sun. The forces on a hill manifested by what you perceive as wind. Look around, not ahead. Look around, not ahead. The peak is just a way to walk at the sun.
Welcome to Translate 2021. My name is Georg Duda and I'm your host here in Berlin between the Red City Hall and the Dome. Unfortunately, only in a digital format and not personally as we initially planned. Together with Orla Smith, I'm welcoming you here and I'm also hoping that Orla can join us and is with us here, and we're together welcoming you and opening Translate.
Hello, hope you can all hear me. I'm Orla Smith. I'm the editor of Science Translational Medicine. On behalf of Science Translational Medicine, based here in Washington, D.C., I would like to warmly welcome all of our conference participants from around the world to Translate 2021, our third in the Translate conference series.
It's a topic that is highly relevant, as we saw in the last year. It's important to translate fast, but there are key challenges that we think we have to face and that will alter the way how we do medicine and know medicine today. Aging in industrialized nations will lead to have more and more patients and less experts who can treat those patients. It's a race for the best talents. Digitalization will alter the way how medicine is done and alter treatment and interactions at all various levels. And a complete new group of therapies, advanced personalized therapies, gene, cell or material-driven, will alter the way how therapy is done and allow the possibility to treat things and diseases in a much different way than it has been ever possible so far. These challenges require novel strategies and new interactions. It will be not so much a separation into diverse rules of key players anymore possible between inventors, regulatory bodies, and investors. We need much more tight interplay. For example, advanced therapies will become a possibility for academia to offer those therapies to their patients in direct need and direct vicinity to the clinical departments and have personalized therapies at hand. Thinking in technology silos will have to change because digital pharma and medtech is more and more merging. Differences disappear, and at interfaces, novel therapeutic strategies are possible. And interactions will change in the way how people interact and how they interpret themselves. All those challenges and novel strategies open fantastic new possibilities for translational endeavors. I'd like to hand over to Ola now.
Thank you, Georges. As Georges has mentioned, 21st century translational medicine faces enormous challenges that must be overcome to translate new technologies, drugs and devices into improvements in healthcare for all. The pace of biomedical research discoveries continues to accelerate, yet translation of this knowledge into concrete improvements in clinical medicine still lags behind. The current COVID pandemic highlights the need for rapid translation to address not only traditional healthcare needs, but also global health crises. The Translate conference is a global forum that brings together key stakeholders worldwide. Translate is not about reporting research findings, but rather discusses the many factors that constitute successful translation. Science Translational Medicine is very proud to partner with the Berlin Institute of Health at the Charité on the third Translate conference. We are the leading journal publishing in the translational space, and our mission is to publish articles that fill the scientific knowledge gaps at the junction of preclinical research and medical applications in order to accelerate the translation of this knowledge into new ways for preventing, diagnosing, and treating human disease. This year's Translate conference focuses on key translational themes. These include metrics of translational performance, acquisition and ownership of individual health data. How can we improve on data reproducibility? The application of regulatory sciences to speed up regulatory approval, and components of successful translational projects and institutes. I will now hand back to Georges for some housekeeping elements about Translate 2021. Thank you.
Thank you, Ola. Just some technical remarks. If you haven't done so, please register. Only then you have full access to all details. All presentations are online, have been uploaded already 2 days ago. If you haven't watched them, watch them. I can just invite you. They are fantastic presentations, and we are just focusing throughout the conference on the discussions. There are 2 consensus papers. Join in and help to make them best possible, use the email button on the page under each of those consensus paper statements. That allows us to track you back and interact with you and come into a communication with you. Ask the panelists by typing the comment feed. The co-moderators will bring your questions to the podium. And finally, we have received translational success stories and translational stories, and we selected 3. They are under challenge at translation. Have a look. Make yourself familiar. There will be a vote in the end about the best of those. Finally, how do you put your question and answer? Do that in Slido. That's a field underneath the screen, and you can ask your questions there. Please use that opportunity. With that, I'm happy to now introduce for the opening statements Professor Axel Pries as Dean of the Charité and Professor Baum, Christopher Baum, as the Director and CEO of the Berlin Institute of Health. Axel Pries, the floor is yours.
Dear colleagues, dear attendants of this year's Translate 2021 conference, ladies and gentlemen, it's my great honor and pleasure to open this year's Translate 2021, albeit under very difficult circumstances, as you all know. I would have loved to see you all in person, having great discussions and taking a short walk alongside the Spree River. All of this is not possible this year, and of course we discussed to skip the conference and postpone it to 2022. But the pandemic teaches us that such a conference of translation is increasingly relevant under the current medical environment. Medical translation is something which is done at the Charité for decades and centuries, and revolutionary medical research findings have come from the Charité from many areas. Rudolf Virchow was the founder of cell-based medicine. Robert Koch discovered the infectious agent of tuberculosis, and Emil von Behring developed blood sera against diphtheria and tetanus. However, these examples also have a lesson to teach us with respect to the complicated and very difficult road to true translation. More than 150 years after Virchow's discovery, modern cell-based medicine is only right now able to start, and with the BRH Center for Regenerative Therapies, which develops innovative cell therapies for patients who receive donor organs in order to keep transplant rejection under control, we see breakthroughs in these areas. The tuberculin developed by Robert Koch didn't really help people against tuberculosis, and only the development of modern antibiotics was able to keep these diseases under control. And as you all know, we have a future where we have to develop much more in the area of resistant infectious mediators. And the sera from Behring weren't available for enough people because it was difficult to produce them. Thus, medical translation is still a very major challenge today, and your conference here is very timely and your contributions are very highly appreciated. It takes an average of 15 years and €1 billion or dollars for a good idea to become, for instance, a new cancer drug. And only around 10% of all projects actually lead to success. We need a translational ecosystem that provides a higher efficiency ratio, maybe even lower cost, and more solutions. Together with the Berlin Institute of Health, which now has become the third pillar of Charité and is devoted to translational research, we are working on creating such an environment. It requires a stable, sustainable infrastructure for researchers and physicians who are trained in translation, are enthusiastic about it, and have the right mindset. we have to facilitate, promote, and reward translation. In order to meet these prerequisites, we need your help. We want to learn from your best practice examples, but also learn about the hurdles and failures, and together we should be able to overcome these failures and lead to joint success. In this context, I'm very thankful to Georg Duda and his great team who managed to organize Translate 21 as a virtual conference, and I'm very grateful to all of you for having accepted the invitation. I'm looking forward to exciting talks, thrilling examples for medical translation, and to fruitful discussions. I wish you all a very inspiring 2 days, despite the digital format, filled with science and innovative ideas. And as we all do, I hope that next time around, at the next occasion, we will be able to discuss in person again and have all these unforeseen discussions and talks alongside the presentations which bring us to the next level of understanding. With this, I wish you a very, very fruitful meeting and very good days. Thank you very much.
Thank you, Axel Pries, for those warm words. And I'd like now to hand over to Christopher Baum, the CEO of the Berlin Institute of Health.
Dear colleagues of the international scientific community, dear friends of translational medicine, spectators and speakers, ladies and gentlemen, there is no question. The importance of translation has never been more apparent than in our current situation of a global pandemic. In these truly challenging times, translation at astonishing speed was achieved by the scientists and companies who managed to develop the first vaccines against SARS-CoV-2. Deciphering the virus genome, developing an mRNA suitable for vaccination against the spike protein, identifying the best dose and mode of delivery, upscaling production for multiple millions of doses, testing the new vaccines in numerous volunteers of many countries, achieving regulatory approval in different healthcare systems, all of these steps happened within only 1 year. Obviously, this was only possible based on multiple robust facts previously acquired over many years in basic research and development, combining different fields and topics, including principles of mRNA vaccination, coronavirus biology, mRNA stability, its delivery, and precision immunomonitoring. Therefore, the rapid success of the new vaccination approaches has multiple parents, and a similar story could be told about the emerging antibodies and most likely in near future even novel nanobodies against SARS-CoV-2. All these cases and stories of speedy translation are based upon strong basic research, detailed technological improvements, individual entrepreneurs who had the courage to start and move risky projects forward, larger companies stepping in for upscaling of production and delivery, and regulatory agencies which were willing to adopt their mechanisms of oversight to the urgency of the global threat in the interest of the individuals at risk. The speed at which all of this occurred is reminiscent of an expression known from classical music, allegro ma non troppo, quick but not too quick. If you look for an example which illustrates that this still refers to a rapid process, I recommend listening to the third part of Beethoven's Sonata Appassionata, which is allegro ma non troppo. Another highlight of the year 2022 was the Nobel Award for the CRISPR-Cas approach to sequence-specific gene editing. To underline the translational importance of this event, 2020 was also the year of proof of concept for the potential of gene editing using CRISPR-Cas for somatic gene therapy of human disease. We are so proud to have Emmanuel Charpentier here in Berlin contributing to our great ecosystem of science. At the Berlin Institute of Health, We are happy to be a major player in this ecosystem, also contributing to the international scientific community with our mission to promote both robust and rapid translation. And this mission is the reason why we are also so proud to host the Translate conference again this year. The purpose of Translate, be it in conventional or digital format, is to connect brilliant people, sound research, strong partners from academy, industry, patient organizations and economy, and regulatory authorities with one common goal: to promote international networking for the advancement of innovation in medicine, to support translation, or as we would like to say at the Berlin Institute of Health, to turn research into health. Finally, with this said, I would like to thank Hans-Dieter Volk and Georg Duda, professors at our institution, and their teams for their contribution to the organization of the Translate 2021 conference, and everybody else involved. And I wish all of you inspiring 2 days with fruitful discussions, exciting talks, inspiring best practice examples, and many unexpected lessons to be learned. All the best for you. Thanks for being with us. Enjoy the conference.
Thank you very much, Christopher, for those warm words. And I would like, with that, directly to jump into the program. The first panel that I'm very proud to have here with us is going to be on the topic of steering versus freedom of translation, lessons learned. The panelists that I'm going to have here with me is as co-moderator Kathleen Chayak from Science Translational Medicine, and we have Angelika Frenzen with us. Angelika is at the Wyss Institute in Boston, where I'm also happy to be an international faculty member, and we met both last February when she started there and I had my sabbatical there long time ago, actually 2 years ago. And she has expertise, over 20 years of expertise in academia and biotech, and she shapes at the Wyss the business development and product development facilities and is responsible for those areas and is incoming Institute's chief operating officer. We also have Robert or Bob Goldberg with us. I know Bob for a long period of time. He's at the University of Oregon and is there the Vice President and Executive Director of the Phil and Penny Ignite Campus for Accelerating Scientific Impact at that university. That is a whole endeavor that was possible through a very large philanthropic donation, and he will tell us about what he has learned so far and how he would like to shape that for the future. We have also with us Louise Jones, who is the expert at the Medical Research Council on translational research. As you may know, the Medical Research Council in Great Britain is focusing on supporting science in general, especially also in basic science perspectives, but has also a dedicated translational research arm. And Louise Jones, with a long-standing expertise in that endeavor, will tell us about how she learned, what lessons they have learned, and how she would like to drive that forward. And the final member of the panel that I'm happy to introduce to you is Hildegard Bühning. She is at the Hanover Medical School. She serves currently as president of the European Society of Gene and Cell Therapy and as scientific secretary of the German Society of Gene Therapy. And she is one of the experts who brings gene therapy into clinical usage, has learned a lot of lessons on those pathway. And the key topic that I'd like to address with those panelists is to talk about how they think, how much freedom is necessary, what lessons they have learned, and how we can bring and learn from those lessons towards the future of translation activities. The idea is we have uploaded all presentations, and if you haven't seen those presentations, I invite you to have a look at those presentations of all panelists and have a detailed look. There are a couple of really very specific points addressed in those. Due to the digital format, we are focusing in the next couple of minutes on the discussion across those presentations, and we'll start with opening statements from each of the 4 panelists. If you would like to ask questions, you can type the questions in, and my co-moderator, Kathleen, will bring the questions from the group of listeners to the panel that we can also address those. We have roughly 550 people registered, so we're very interested to hear about the questions that come up from the panelists. With that, I'd like to introduce Angelika and ask her to briefly introduce herself to the panel and to our listening auditorium.
Yeah, good morning. I'm very happy to be here. At least here it is still good morning. I'm calling from Boston in the United States. Very happy to be here. I am German, as some of you might pick up from my accent, so I'm closely related to where this conference is coming from. Then transitioned for my postdoc to the United States, where I was working at Harvard. I'm a chemist by training. And after my postdoc, I had the privilege really to be asked to join a very young startup company founded by 4 postdocs. And I spent 14 years in this company, all the way through to a commercialization of a therapeutic, Linzess in the United States and Constella in the European Union. I joined another smaller startup again for another 4 years, brought a product into Duchenne muscular dystrophy into phase 3 clinical trials when the opportunity came to me to join the Wyss Institute and very actively think about translation. And translation in an academic setting and in a very exciting ecosystem in Boston. So I'm very, very excited to share my thoughts on this, but of course, I'm not researching this field. I'm more part of the ecosystem and therefore happy to share my thoughts.
You can directly go into the points if you would like to address them more in depth. Do you have the list of your questions in front of you or the take-home message points? Actually, otherwise I would ask that the take-home messages slides are presented for Angelika. Yeah, thank you. Do you see them, Angelika?
I do not see it, but I can put it up on the side.
Oh, I'm sorry.
No worries. Okay, so my slide that I'm presenting is my thoughts on the translational ecosystem that I have experienced. We're calling it innovation ecosystem. The Wyss Institute is a biologically inspired institute, so it makes sense for us to call it an innovation ecosystem. So what we are thinking is necessary for this ecosystem is a localization of capital talent around certain hubs, right? And for us at the Institute, that's our academic contributors who generate high-risk new ideas and who we provide through our funding freedom to operate in a wide range of different technologies, from synthetic biology to engineering, chemistry, drug repurposing. So there's all kinds of fields that are really pursued very freely. And we hope that localization of capital and talent around these hotspots is really helping us to solve not only healthcare, but also sustainability problems. And then what we're trying to bear is a diversity of functions. We believe in a convergence of disciplines. You will find at the Wyss that a lot of times when you solve larger problems, you might want to bring in chemistry, biology, software engineering, hardware engineering to bear to really come up with a solution that is impactful. So the diversity of functions and the conversion of disciplines is another aspect that we think is key to not only strong innovation, but then also translation. And then once you want to generate a lot of ideas, but also pick the best ones that are most suited for translation and solving a larger problem, then I think the community, the collaboration, the strong community connections is another essential part of that ecosystem. If you want to throw out the not so fruitful ideas. You want to focus on the best ideas. You want to connect, and you want to make sure that people share ideas, approaches, and ultimately a pipeline in a community. You have to have trust in this community. And I do think we're seeing this very strongly around the vaccines right now and about the COVID data. If you don't trust the data, if you don't trust the community that tries to solve a problem, it's hard to solve a large, complex problem. And so we are very much hardworking or very focused on the community building at the Wyss Institute within our academic collaborations, industry collaborations, or collaboration with venture. And then I think, again, in the context of community, it is important to let it self-assemble. You cannot put on top an idea that a certain person has to work with another certain person to solve a problem. I think innovation, collaboration, and then ultimately product development happens best when the right players get together with a lot of fluidity and self-decision. And so, in summary, we do think at the Wyss, and that's how our model is designed, we do think that each of these aspects are very important to the innovation ecosystem and to translation. And if you take one of them away, either the resources or the fluidity or the innovation hotspots or a really strong community, you will weaken this overall ecosystem and therefore weaken the translational effort.
Thank you very much, Angelika, for that insight. And as I said, if you want to hear more about how Angelika developed those points in her talk, have a look at the talk later. Now I'd like to invite Bob Goldberg to give us his take-home message statement and eventually tell a little bit more about yourself. If you could start, Bob, the floor is yours.
Thanks, Georg. I'm Bob Goldberg. I'm a bioengineer. I work in the musculoskeletal regenerative medicine space and have had the opportunity to start 6 companies over my career, and then was at Georgia Tech for about 22 years, in which case I was part of a very thriving innovation ecosystem that was well established. And then a couple of years ago, I had the opportunity, as Georg mentioned, to move to the University of Oregon, and with some very significant resources from Phil Knight, who is, I think most know, a very successful entrepreneur in his own right, having been the founder of Nike, provide us resources to set up this campus. And you can see the first building here behind me. And so it's really an amazing opportunity with these resources to set up, design and develop a new innovation ecosystem sort of from scratch. In contrast to what Angelika just spoke about with the Wyss Institute. So my take-home messages, which Georg, can you confirm that they can see those?
Yeah, I hope that, can we see the take-home messages from, yes, they are on the left-hand side.
Okay, so very good. So, I think with starting a new ecosystem like this, there's advantages and disadvantages to that. And one thing that's very important in starting something new, and probably in all cases, is really to clearly articulate a mission to translate. In our case, the mission of the Knight campus is to accelerate the cycle of translating scientific discoveries into societal impact. And I think that's really important because as people come into the field, students certainly and most faculty early on, they're very driven by developing, papers and grants and so forth, but they really want to make a difference in the field. And I think sometimes we get lost in counting things and maybe lose perspective on the bigger picture of why we entered this field in the first place. And so having a very clearly articulated mission with a strategic plan behind it really helps to attract the right students, faculty, donors, collaborators, companies, and investors to your endeavor. The second take-home message is really about the team, and I think that's probably a theme you'll hear throughout this. It turns out much literature shows the importance of the team and having particularly past experience on the team in entrepreneurship and interdisciplinarity. And in fact, those two aspects of team are even more important than things like having local resources like venture capital. We all know the most interesting and innovative solutions are often found at these interfaces of traditional disciplines, and that's effectively how we solve tough problems. And so, I have the opportunity with starting something new to really recruit people that are interested in doing this, recruiting talented faculty and students, but also, of course, very dedicated staff, engaged companies and mentors who have been through the hard knocks of starting companies. And that will help entrepreneurs avoid the pitfalls and make connections they could not otherwise make. The third take-home message is really to create the time, space, and incentives that are appropriate for faculty to pursue translation. You know, there are very real economic pressures at universities to teach more, since there's reduced government support, at least in the United States, which means that tuition dollars are absolutely critical. We're fortunate in the Knight campus to have some philanthropic support that makes us somewhat immune to budget cuts from the state and therefore can provide faculty with teaching releases, for example, for entrepreneurship. Not every institution has a Phil Knight behind it, but most have alumni who have had success in business that may be willing to help in this manner. We've even built into our tenure and promotion incentives, incentives that are aligned and not necessarily to just form companies after you're an old and tenured faculty like me. So if our young faculty form companies, then they have the ability to get credit for that, along with research service and teaching. The fourth take home is really to integrate into your curriculum academic programs that teach science students about entrepreneurship through partnerships with the business school. This is a really terrific mechanism and model to have great students that can go off and perhaps even be the CEOs, but certainly key people in companies that are formed out of the academic environment. And finally, I think a final take-home message is that we really need better tools to measure the diffusion of knowledge and translational impact. We often think of these things as linear processes or perhaps, you know, an impact cycle. And really that's not adequate for capturing the actual impact of knowledge diffusion from academic research. It's not so hard really to track how discoveries, patents, and papers perhaps result in a single commercialization venture, but those ideas may have sparked other innovations elsewhere. And we need to have better and more sophisticated tools to capture that impact and not just but also simple ways to communicate it to non-science and non-business people.
Thank you very much, Bob. I will come back to those points in a minute. And now I'd like to ask Louise Jones to give us her take-home message and her experiences sort of in a nutshell perspective. And also there, probably one word before, Louise, you're starting. Next, the MRC is here, a very valuable contributor. I should have mentioned on the very first moment when we started the conference that also the German Research Foundation is heavily supporting that event. And we're very proud also to do that together with the German Research Foundation. But now on purpose, the perspective from across the channel, which is so important those days, especially in this year. So, Louise, the floor is yours.
Thank you, and thank you again for the invite to do this. And I'm sorry that we all can't do this in a more personal, face-to-face environment. So my name is Louise Jones. I'm currently the head of translation at the Medical Research Council, which is one of the it's part of UK Research and Innovation, which includes all the thematic research councils. Sort of the perspective I'm going to bring today is really the nuts and bolts of funding. So not necessarily as an entrepreneur. I grew up through a very traditional academic career, a lecturer in pediatric oncology before I decided to sort of change gear. So the latter half of my career has really been focused on that translational research and the management of that research at the programmatic level to create systems that enable strong translational research. And I've done this at both at MRC, but also at Cancer Research UK before that. So in terms of my take-home messages, I mean, the first thing to say is I started with backwards forwards. So first of all, I took you through an evaluation, and this was very much to show that actually we do have outputs and impacts which show that dedicated translational research funding works. It works well. We have some outputs and impacts, and we can talk about some of the metrics. Some of it is bean counting, for want of a better word. But equally, we've found other ways by which we could assess spillover, and that could be through enhanced business engagement, collaboration, be that through porosity of postdocs moving from that project into the partner organisation, the partner company. The other thing I wanted to say here is we looked at translational research over a 10-year period of time. And during that time, it was actually very hard to determine patient benefit. And for me, this just highlights the fact that translation takes a very long time. And even if you have a good headwind, you need lots of dedicated funding, and you need the continuity of funding as well to do that well. As Bob pointed out, and as we all know, that translation's not simple, it's not linear, it's very complex and needs a lot of iteration. And this means you need different types of funding and different tactics to support translation at very different time points during that process or that translational path. And again, hopefully I was able to show you some of the vehicles that we actually use for that. Again, looks like I've been copying Bob's sort of talk in some respects. My third bullet was around that team science. And again, critically, if you were to take one key take-home message from a strong translational project that is peer-reviewed, it is having the right team. If you don't have clinical, you can have an amazing transformative technology, but without a clinical lead, it's not going to go very far. If you're doing a clinical study and you don't have stats input, again, you're going to find that isn't going to go terribly far at all. So again, really thinking up front what that team is and actually where you're going to go. The peer review process, actually, some people will find quite laborious, and I can understand that, but it's also incredibly useful as well. And what it can do in translation is really help and support those people who want to engage in translation. For me, translation is very much about maximising discovery science. And for us, we do that across the whole of the UK. And so using peer review, be that through our brilliant national and international reviewers or through the dedicated panel of experts, it means that through feedback and support throughout the whole of the lifespan, from a project idea all the way through to project award and development, again, they have a key part to play to upskill and really help people understand how to do translational research. And my final point is, and I think it's been made before, that the projects will go wrong, they will change, they will need to change. And actually it's the balance of that flexibility to enable, a change that is valid and rational, the freedom, the flexibility to make that change without being too constrained, while actually still keeping a project on a development path, on a route towards either patient benefit and or commercialization. So projects will fail, and that's fine, and Milestones will help you reach that goal. So on that note, I'll stop there.
Thank you very much, Louise, for those insights. And you can already feel how much experience is behind those words. Now I am very happy to look forward to the statements from Hildegard Bühning. She's working at the forefront of an area where everybody of us is really expecting substantial changes to the way how we do medicine today, the gene therapy arena, and bringing that forward. And I'm very happy that you're here, and we're happy to hear and listen to your take-home statements.
Thank you very much, and thank you very much for the invitation. And let me say again, you said, my name is Hildegard Bühning. I'm from Hannover Medical School, and there I'm focusing on adeno-associated viral vectors and optimize them for cell and gene therapy. And actually, I studied biology with the idea that I want to make gene therapy or help a bit to make gene therapy a clinical reality. And this, on one hand, I try to do with my research, but on the other hand also, and this I think brings me already to my summary slides or my statements, by engagement in societies. So on one hand, internationally, it's a European Society for Cell and Gene Therapy, and nationally, it's a German Society for Gene Therapy. Because what I believe, what is required is that we really have a strong scientific network. And we have seen this with the COVID pandemic, what really this can bring. We have to define and agree on a target to work on. We need to have a financial commitment, and what we heard already in the welcome speech of Christopher Baum, we also really need to see the contribution, and we have to strengthen basic and preclinical research, because this is really feeding up our pipeline and helps us really understanding if something was wrong or what we have to work on. We have to intensify the interaction with the regulatory bodies. It's quite excellent already, but the back and forth will help on one hand the science, and on the other side also the regulatory bodies to help us bringing the new therapies really to the patients. And then we heard this already from our colleagues. It's absolutely important to engage with the patient organization to see really what they would like to see from us. And on the other hand, only to really take them into our ideas and that we really can explain what we want to do. And we have really to think about and help our next generation scientists to really fascinate them for the project and the targets we are on and really teach them. And teaching means also that we have to really educate our lay public so that they really understand what we are doing, why it takes sometimes a bit longer, and on the other hand, that they are not frightened of what we are doing. Thank you. I'm looking forward to the discussion.
Thank you very much, Hildegard Bühning, for these insights and statements. I'd like to start the discussion here within the podium and invite you also to use the question and answer feed to bring the questions to the podium. I'd like to start with Angelika. The fantastic story of the VISUS, it has been established, it has built a spirit, it has built a team, and you pronounced that also, how important the team and the fluidity of the system is. That implies to me also that you need quite a bit of freedom to realize that. Yet there is, and I hope I don't say too much here about like sort of secrets, but there is a little bit of certainly an interest of institutions like Harvard to control entities. So you have on one side the interest of large institutions to control, especially also the translational endeavor, especially towards medicine, because you're also eventually liable about things that go from there. And on the other side, you need the spirit and the freedom to actually create new things, get startup companies realize you need to be fast, probably sometimes faster than regular universities are in processes. Yet there is another example like the Broad Institute close by, who has a little bit even more freedom than the V's eventually. So how much freedom would you like to see, would you wish if you could? And how much freedom would you recommend others to really build into their structures? For example, Bob, when he's still, he's probably already has built those elements, but how much freedom is necessary to get that really, keep that and get that rolling?
Yeah, so the Wyss Institute is an interesting mix in this regard. We have a philanthropic funder who provides us some degree of independence and freedom. And then we are associated with Harvard, and I personally see that as a positive because we get a lot of incredible talent into the Wyss, who then with a little bit of funding and freedom, or sometimes a little more funding or freedom that we provide, can really come up with the innovative ideas. So to some degree, I feel the Wyss has the best of both worlds. We have access to talent. We have our own funding avenues, not only through Hans-Jörg Wyss and his philanthropic donation, but through grants and the grant agencies we go after to get additional support. And we're trying to combine both. A lot of times, I think the ultimate innovation and translation lies in the connections you build. And so you can build a framework that has impact based on, high risk innovation and a lot of freedom you provide. But then also we take advantage of a lot of the infrastructure at Harvard and some of the guidance. We have a lot of colleagues across Harvard that help us to weed out the ideas that we really want to pursue. So I haven't seen that tension as much. Sometimes, of course, we face a little bit of bureaucracy. Harvard is a big organization and has to keep things, for example, conflict of interest considerations in mind. But overall, I do think we have found a little bit the sweet spot to be part of a very important organization. At the same time, have some freedom to pursue, not only with our collaborators at Harvard, we also have collaborators at MIT or Tufts or the hospitals to pursue the ideas we really want to pursue.
And following along those lines, is it not key to keep that philanthropic freedom that you have? And how do you envision to build that if there is not a fourth gift going to come up? So how is your strategy to keep and maintain that freedom in the long run?
Yeah, philanthropy it's a large component of what we need to think of, right? So the Wyss was built on a philanthropic donation and has developed its own mission and strategic plan, as Bob was highlighting how important that is. It will always play a role, as do alliances with our partners. We have, for example, started last year a very interesting alliance with a venture partner, and I think I outlined that in the call. And there is the important access to infrastructure and support, and quite powerful support in Boston through Harvard. So we need both, all three items, right? The alliances, philanthropy, as well as support from Harvard. And I think that brings the bees forward and brings the translational technologies forward.
Probably handing over to Bob at that point. You have such an even substantially larger donation scene. You build something completely new there in that environment, and certainly you need also that freedom to get forward and be then successful and show that with the various models that you also explained and illustrated in your sketch. How free are you with that philanthropic approach, probably also in comparison to your previous time at Atlanta, where also there is a big local industry eventually donating into that concept, but probably in a different role than you probably have seen now in the Knights campus in Oregon. If you compare those two situations, obviously you will favor the current one. How much freedom do you need from your sponsor and with your sponsor in both directions, basically?
Yeah, I think, as I said, I think it's increasingly important because, at least in the States, with the less appreciation for the economic impact and innovation, that the universities have really been that engine, and there's less support from the states for higher education, and so that's put a lot of pressure and less freedom on the rest of the university. So having, in some ways it's similar, I think, although at different stages of development with the Beast, where we have this partnership with the university that is beneficial going both ways, but we do have some freedom. I think maybe the one thing that I have that's a little bit different is that the faculty I'm hiring actually have their tenure through the Knight campus. And so I have a little more freedom to be able to create space in their time and in their tenure promotion criteria for them to do entrepreneurship. And so we're certainly, taking advantage of that and making sure that they have the time to do that.
Louise, probably along the line that you sketched, you are in a different setting in your translational arm of funding to most of the other elements in the MRC. To be able to do what you are doing, did you need to install a new type of also freedom within your institution, because you need a different interaction with your people? For those who probably haven't listened to that, to the talk yet, Louise is basically, once projects have been brought on board, they are really accompanied throughout the process. So you don't just support them and then you review them 4 or 5 years later. You basically continuously are in an interaction with the project. So it's a different culture. How easy was that, bringing that culture into your funding organization?
It's a really good question, and I think the first thing to say is that the MRC it predominantly funds discovery science, and actually translational research is an output of discovery science, be it serendipitously or indeed through intent, as well as being supported through very directed translational funding. So, our translational funding is run a little bit differently to our discovery mode, response mode funding, in the fact that we do monitor progress of the project set against the milestones that the principal investigators themselves put in place, obviously under guidance. And this means that we can help and guide those applicants through that translational path to ensure that they meet the goals of that project, from the outset. So in this way, it is quite different to other standard response or standard grant funding, where perhaps the money is awarded and then there may be sort of it's next looked at when the next set of funding is perhaps reviewed, be that through high impact publication, etc. So. What this means is, in our dedicated project funding, if you have a sort of a preclinical project and it's met its milestones and coming to the end of a project, that means they are able to then come back for a second tranche of money that may take them through that early clinical sort of next step of the translational path, that first in man, which then might give them, as I say, having de-risked the technology to a point that they are then able to attract either VC or other funding from other organisations to help that journey downstream. I mean, the UK has I have to say, a buoyant landscape and ecosystem. MRC, while a major funder of medical research in the UK, is also supported by, you know, a strong charity sector, albeit one that's obviously taken a little bit of a knock recently with the pandemic. And on the other side, sort of through NIHR, to support that downstream public funding into that later phase clinical trial, you know, sort of elements of translation. So I think it's a balance, because if you have too onerous a reporting and monitoring schedule, then it can actually, you know, it can impede translational research as well. And as a public funder, while we need certain levels of assurance, it also means we have a slightly more, we have a flexibility to support things that actually might only benefit a very, very small patient group. Say, for example, a very ultra rare disease where, again, it's unlikely to be supported perhaps through company venture, etc. So it allows us a great deal of flexibility. So, but I'll stop there.
Hildegard Bühning, are we already sort of with gene therapy? Is it too early to ask the question about venture capital funding that we need, and we have a different culture? I mean, don't need to explain that. The culture in Germany is different in the sense we don't have the Kendall Square atmosphere directly in front of Hannover University Hospital, and we're basically but on the other side, we're really positively funded by DFG and Federal Ministry of Research and Education. How do you see that role of philanthropy and venture capital, especially probably the area of gene therapy? Do we need that more, or is that just probably the way how it is organized at the moment, giving enough freedom to move those topics forward?
I mean, it's an interesting question, because if you think a little bit backwards, when we had the unfortunate cases of leukemia or Jesse Gelsinger and so on, there was somehow a fear of venture capital to invest in the concept of gene and cell therapy. And this has now changed. So there's trust again that the concept really is promising, that the concept will change medicine. And now the issue is, how can we really bring this to the patients in need, and the patients here in the Western world, let's say, but also that on a more broadly scale. And so as you have we all have seen now the interest from industry coming back to gene therapy, and this really helps. This helps a lot. Because as I was already saying in my summary slide, it's a long way until we have something which really is in question then coming forward to the patients. We need funding there, and it's good that we have the German Research Foundation helping us here. But when it really comes to clinical trial, there is a lot of money needed. And also then when it comes to manufacturing places and so on. So I think here a close interaction with pharma industry is required, but it has to leave us a sufficient freedom really to do our science. And I at the moment I see that this is indeed the case.
If you have also questions that you would like to pose to each other, feel happy or feel invited also to do so. We have some questions from the podium and coming in, or from the auditorium coming in, that refer to basically both, to some degree, to basically most of you. What vetting process was used by your Night campus was basically one question going to Bob, but I think it also fits perfectly to Angelika, and it could also be adapted to the MRC perspective. What vetting processes were used for you when you build up those instructions or those structures to boost translational endeavors, and to consider many of the existing institutes that have seen certain successes and certain shortcomings here and there. And I know that Angelika also readjusted some elements at the Wies also with a certain reasoning. So what are the lessons, the key lessons probably learned? I know that's the whole talk of you, but probably it's coming here in sort of as the key questions in the flow. So that's why I'd like to bring that also into the discussion. Can you break that down a little bit further?
Answer me. that's a big question to give a short answer to. So I think the key lesson is that you want to focus on staff and faculty that have at least in some degree for the research and end user focus, right? So the idea of you developing technology with an end user application in mind, and you create the robustness of the technology around it, is a central idea to the Wies. And so that's something when you refer to that, some of it, we might have fostered it even a little bit more in the last 2 years. And so that has to kind of be pervasive to the whole, institute. If it is the translational arm of research, our faculty has their own labs still at the academic institution, and that's where they might do very foundational research. But the projects that come into the Wies a lot of times are judged by, do they have an end user in mind? And is the technology then validated towards that end user need? And is the team able to put specifications in place to say, these are the things we need to meet to make this, to bring this technology forward? And I do think that has to be pervasive to the thinking. And then, not unlike what we have experienced in startups, is then the next step is to set up your go, no-go experiment and make sure that you kill the ideas that don't meet the specifications. So that's where then the interface between academia and what we have experienced in industry comes in. That's where the community comes in. That's why I emphasize that, so that if you kill an idea because it doesn't quite work, nobody takes it personally. It's a matter of, pursuing a pipeline. And so I'm going to stop there and give it to Bob, because I'm sure he has some thoughts on this too.
Yeah, I think Angelika makes really good points that ecosystem's the right word, because there's so many different elements to it. And if you're missing any of them, you have a weakness that you have to figure out how to address somehow. And so, Wies has incredible resources that we're just building up. So we have to get to that point, but having those resources available and facilities and so forth for people to do their work is really important. I have maybe a unique advantage in that I'm starting from scratch. I don't have to come in, and I'm not introducing something new to an environment or to a set of faculty. I get to recruit the faculty and the students that are attracted by the mission that we've put forth. And so that has some advantages into bringing in like-minded people. And then one of the strategies certainly that we're using is to really provide very low barriers to get started. Almost no questions asked for just the initial bolus of funding to go out and get incorporated and do some consulting and get the company started. And then, bringing those people, those entrepreneurs, those budding entrepreneurs into contact with people that have done it before. That really, I think, is maybe the number one important factor. There's the Creative Destruction Lab, which I talked about in my talk. One of their major hypotheses is that innovation happens everywhere. If you're an alien and you were looking down on Earth and you were looking at where things actually get commercialized, there'd be these bright spots. But if you are actually looking at where innovation happens in research, it happens everywhere. And so why is that? There's a concentration really of entrepreneurial judgment or knowledge or experience in some of these spots that help to move things forward. And so I think that's really an encouraging message, because if we can facilitate that sort of interactions and that sort of contact with experience at our own institutions. And of course, the Wies Institute has that right there in Boston. That's one of the hot spots, but Oregon is not. And so that's a challenge I have to help to do, and then to connect, the people with that type of experience.
I think those are both very, very valid and very important points, and thank you for being so open. Louise, how do you do that in the MRC? Because if that team building and that team experience is so important, but you yet fund every team individually, it's all over Great Britain. So is there any means that you ever thought about how do you support that team building feature with a funding as a funding agency, or is that just something that can happen at locations?
No, I mean, it's a really good question. And I think the first thing to say, when we did the evaluation, it's very much looking at the landscape and kind of what was needed and what types of intervention. And one of our council members asked us, after analysing all of this stuff, 10 years, what's the magic recipe? What can guarantee success of translation? I'm not sure I have the answer to that, but obviously we all know the key elements that have been highlighted, so I won't go over them. I mean, the one thing that we do to really enhance that team culture is that we have a program called Confidence in Concept, which is devolved out to research organisations. So any university in the UK can apply for it with a clear strategy. And if they've got a really good translational strategy and can show us they've got a pipeline of innovation, then we will give them, some money, and this can be quite a substantial amount of money, up to 800k in a year. And this money they can use flexibly to support awards that they choose in their institution, and that they can be collaborative with industry, for junior. Sort of early stage career fellows or kind of whatever. We give them that flexibility. And actually, so what we found is that really, really engenders a culture of translation in these research organisations, because it allows people to build the confidence, generate that critical data set that then can be used to sort of apply for more formative funding, for want of a better word. So this, for me, is a way at a small scale, small amounts of investment can actually make a really big difference and set that individual or that research organisation on a path to actually doing a lot more in that translational space.
Thank you very much. We basically have touched a lot of a number of those aspects, certainly not all of them. One question that really burns to me, and probably just a quick feedback from Hildegard Bühning. We haven't, and basically I could ask that question basically to all of you. We talked about the medical expertise, we talked about the technology expertise, but we didn't talk at all too much, not really in depth, about the business expertise. And we talked about team building, and part of that team is always that business perspective and competence. How easy or how complex is that, finding those expertises also now in gene therapy, vector strategies? Are we already at that stage that we need those expertises, or are we too early for those novel therapies to already think about the business perspective of that vision?
It's ongoing. I mean, this is an absolutely active field. So a lot comes from academia with startups and ideas, really, that they try to bring their own ideas up to a gene therapy product, but also teaming up with bigger farmers. So it's all up the screen.
But do you have a business school, for example, in Hanover that you team with, where you get entrepreneurial competences on board when you build new teams there that also require a business competence? Or do you think it's rather the upscaling to pharma that's the pathway?
No, so in Hanover, we don't have a business school hereby that we speak to. But I mean, if you really look into the scene, then you see that automatically we do have these teams which are building up in cell and gene therapy, where you have really teams from the different areas. That is what I mean with a scientific network, and a strong scientific network. And you need all the expertise from different areas, and this is a business needs to be there, of course. I mean, we heard this from the colleagues, and it's in gene therapy, it's there already.
We have, I'm sorry, we have to stop here to get to the next podium discussion. We have fantastic questions still from the floor, and I'd like to at least bring one here, which we will not be able to due to time to ask, from Glenn Prestwich. Can each presenter tell how the institution's translational programs bring market pull into the ecosystem versus technology push, reverse pitches, and how you basically make a push and pull scenario reality? That's something we probably need to address in the following talks and discussions. Thank you very much for your contributions and for that panel. And I'd like to hand over to the next panel and to the next following discussion. Thank you very much, all four of you. Kathleen, are you?
Hello. Hi, I'm Caitlin Chaika, and I'm the Associate Editor at Science Translational Medicine. and I am handling manuscripts in bioengineering, musculoskeletal, and cardiovascular disease there. I'm very happy to be the moderator for the next session, Data Science and Stewardship. Welcome with my co-moderator, Georg Duda, from the previous session. We have a very interesting panel today, which will be discussing large data sets, the use of digital medicine, wearables. We have more data than ever before, but with this comes issues with how to analyze that data, how to handle it, what are privacy and governance concerns, how can we ensure that results are reproducible and ethically performed, how do we set up clinical trials to best meet those needs that we're searching for with these massive data sets, and really how do we do this in real time, where we have technology outpacing regulation, outpacing our analyses methods, outpacing our traditional experimental designs. And so we have 4 wonderful speakers with us today from different areas, and I'd like to give a few minutes of introduction to each. We have Jennifer Goldsack, who'll be joining us from the Digital Medicine Society. She's the co-founder and executive director of DIME, which is a non-profit that advances digital medicine to optimize human health. Her research focuses on applied approaches to the safe, effective, and equitable use of digital technologies to improve health, healthcare, and health research. She's also a member of the Roundtable on Genomics and Precision Health at the National Academies of Science, Engineering, and Medicine. Next, we have Dr. Christoph Roncalli from Berlin Institute of Health, where he is the chair for clinical translational science at Charité Berlin and the founding director of the Common Clinical Study Center of Charité and the Berlin Institute of Health. He's also a member of the Council of Experts for the Assessment of the German Healthcare Development that advises the Federal Health Minister. And he was previously the head of the Department of Translational Oncology at the German Cancer Research Center. Our third speaker, we have Christine Manta from Electra Labs, where she's head of research operations and leads the development of the Atlas platform to facilitate the evaluation and deployment of connected sensor technologies for remote monitoring in clinical and research care. She has prior academic research experience at several institutions and medical centers, and she also supported the Clinical Trials Transformation Initiative systematic review of feasibility studies using mobile technologies in clinical research. And last but not least, we have Dr. Andrew Morris joining us from the University of Edinburgh, where he's Vice Principal of Data Science and Inaugural Director of the Health Data Research UK, which is the National Institute for Health Data Science. Prior to this, Dr. Morris was Dean of Medicine at the University of Dundee and Chief Scientist at the Scottish Government Health Directorate. His research focus is on informatics to chronic diseases, very broad. So I hope you've had a chance to look through each speaker's pre-recorded video lectures, and I look forward to a lively discussion among everyone. And without taking more time, I'd like to hand over the first 5 minutes to Jen Goldsack, who will speak with us about data governance and privacy in the age of digital medicine, covering her 5 take-home message points from her lecture. And before we do that, I'd just like to remind everyone to please type your questions into the slide chat feature that appears below the live broadcast, so we can answer those during the discussion. So over to you, Jen.
Fantastic. Thanks so much. And thank you so much for including me today. So that's right. I'll try and set our panel off on the right direction, stick to 5 minutes as I make my 5 points. So I think one of the things I was asked to speak about was around these concepts of data governance, sort of privacy, so on and so forth, and how they pertain to data rights. And the first take-home message that we should linger on for a moment here is, as we think about data rights in the digital era of medicine, I'd strongly recommend, and hopefully we can have some good discussion around, thinking much more about data governance than data privacy. It's perfectly easy to keep something private. We can put it in a manila folder, as we always have. We can lock it in a file cabinet and it can never see the light of day. I think the opportunity afforded by the digital era of sort of healthcare is that more than one individual can access data at the same time. There are lots of ways that we can benefit from the insights in an individual patient's, participant's, or person's data. So rather than think about privacy I think we would all benefit from establishing strong and robust governance procedures that allow every individual to make decisions around how they share their data. They may choose to only share it with their clinician, for example. They may choose to share it with family and friends. If they have a rare disease, for example, they might be highly motivated to donate their data and share with appropriate protections that information with a research community. Some folks may be willing to share anonymized versions of their data to establish and inform and improve a learning healthcare system. If we focus on data governance, we start to think on strategies and fit-for-purpose approaches to keep people's data safe and allow them to authorize and decide how it's shared, with whom, and for what purpose. And I think that takes us into my sort of second takeaway point, which is about the difference between something like data governance and security. And I think that often those two things are confused, so it's worth pausing there for a second. When we're talking about the security measures around any sort of data, but digital health data in particular for today's discussion, we're really thinking about measures in place to prevent the unauthorized access to data. When we talk about data governance, we're talking about what are the strategies to allow an individual to make decisions around how they authorize the use of their data. So it's important that we don't conflate security practices and measures with data rights and governance. There's also an awful lot of data of all sorts flying around today. So my third point is around defining digital medicine, which is the evidence-based use of digital tools and practices in the support of digital, in the support of medicine broadly, whether that's patient care, whether that's research, whether it's public health, whether it's health promotion. There's an awful lot of information we could, for example, scrape from your search history, and we might even be able to infer some people have health conditions. I wouldn't define that as digital medicine, although there's a lot of promise there. We really need to focus on the evidence-based use of these tools and use that as a stake in the ground and insist on evidence as we start to integrate the era of digital technologies into patient care. I'd like to do a plug as my fourth point for my organization, the Digital Medicine Society. We are a non-profit member organization dedicated to advancing the safe, effective, ethical, and equitable use of these technologies. We have a huge repository of information and resources for folks already at dimesociety.org. We also welcome anyone who's passionate about our mission to come in and join us. There's a lot of work to do, and we are better when we have more experts at the table. And finally, again, perhaps something provocative that we could revisit during conversation. As we move into this digital era of data, as we know that our data is no longer in that manila folder, in that locked file cabinet, it occurs to us that we should think about how we define patient safety and potentially even redefine it. This idea that there are potential harms that accompany misuse of data and information, harms that didn't exist before prior to the digital era. So hopefully something we can get to in questions is my final point, that should we redefine patient safety in the digital era in health? And with that, I think I stuck to time, and I will hand over to my fellow colleagues.
Thanks so much, Jen. And I do hope we have time to talk about that patient safety at the end. So next, let's hear from Dr. Christoph Ankaye from the Berlin Institute of Health, who will be summarizing his main points on his lecture, How to Boost Translation by Data Sciences, Cancer as a Role Model. Dr. Ankaye.
Yeah, thank you very much for your interest in our work and a little bit of the opinion. I have tried to summarize some of our experience, also trying to maybe look at a couple of novel or then well-tested approaches for other areas. So if you are looking through my talk, my take-home messages would first revolve around first principles. Basically, the idea that currently we're looking at data processing very much from a standpoint of an established system and trying to convert an established analog system into a digitalization without, however, looking at why we're doing that. So a lot of the problems that we're currently seeing are revolving around stakeholder discussions, danger discussions, all kinds of theoretical discussions. And I would hold that sort of like a lot of the technological companies who have developed surprising things. I showed the rockets of Elon Musk's SpaceX not only flying up to the skies, but also reversing back backwards down to earth. There may be very simple solutions to our perceived complex problems if we take a first principle approach. So basically thinking about what it is that we really want to do, and what are the elements of our current thought process that we have. We have a very strong kind of molecular medicine. We have a lot of good results from molecular biology and immunology in cancer, for example, but also in other diseases. However, the health system completely fails to form them into a complete solution, or even being able to track the results of the treatments that are performed under these auspices in the future. And so we need to take a strong look at the single elements of what is available to us, all this progress, and some of the areas where this progress is hindered by something that allows us to not process that information. And then finally, how can we take those elements and recombine them into something that is both novel and helpful? And I think one of the main things is that in other areas of society, we have done that by applying a vision zero. Like in traffic safety, in airplane safety, in workplace safety, for the last 50 years or so, we have said as a society, we are not tolerant of avoidable cases of fatalities or deaths from the societal activities. And therefore, I think we should be thinking about data processing, about translation, and about medicine in a vision zero approach that tries to apply first principles. What are we trying to do? We are trying to avoid unnecessary death cases. And so we should return to that thought process on what does it actually take. We need to, I gave an example, we need to simply find patients with blood in their stool in order to prevent colon cancer. This is not a stakeholder analysis. This is a very simple problem. It's hard, but it's simple. So the second point that I have is basically, and I'm including myself as much as anyone in this, where am I part of the problem? We have an approach to digitalization and translation, sort of that somebody has jokingly called the partnership passive. The living room needs to be cleaned. Someone should be carrying out the trash outside, right? Not me. Somebody else should be doing it, right? So basically, the question is, where am I part of the problem? Am I really sharing my data? Am I really processing my data in interoperable formats, or am I just participating in discussions that interoperability is the next big thing and that everybody should share? But in fact, I'm thinking, well, I might get scooped, and so really let's wait until the third publication from that very interesting small data set that we're currently having. So I think a lot of the issues that we're seeing around translation and the limitations thereof do not have to do with actual, but more so with perceived obstacles. I think cancer, and then also, of course, the pandemic, that's my third point, is a good example in what we could have known. Like, what could we have known if our health system had all the information readily available in a digital format? What could we have avoided? What could we have done if we had pursued the formation of a vaccination against SARS-1, where even the spike protein shares 93% identity to the current SARS-2 protein. Cancer is also a very good example. How much better could we be if we understood that even in complex molecular or immunological phenotypes, every doctor could look up the last 10 or the last 100 patients in the country, how they were treated and how they fared. And so I think we have a lot of examples of things that, again, are a simple thought process, very hard to do, but I think both the pandemic, but also cancer can serve an example for the rest of the health system. Therefore, the conclusion is very simple. I think in modern day medicine, if you don't have good data, you can't do better deeds. Patients have, this is not an ivory tower kind of research endeavor, but in fact, a lot of patients, especially in cancer, have to come to depend, their life has come to depend on complex data that is stored in computers, that is analyzed in computers, and that where the naked eye and the hand of the doctor is of very little use unless they have available that data more or less in real time. And then with the mantra, this is not free of charge. So with the mantra from, I think, economy and social sciences in economy, where the term has been coined that people are not stupid, life is hard. I think we should think about that these problems are simple, but they're hard. And we need to give it a sufficient amount of effort, and we need to give it a sufficient amount of resources to address them. But we also need to, I think, find the right balance in evaluating human life compared to such things as data safety, innovation in medicine, and perceived safety measures that are currently sometimes keeping us from saving our patients. So I'll be very interested what your opinions are on some of these statements, and I'm looking forward to our discussion.
Thank you so much, Dr. Von Kaye. We'll now move on to Ms. Christine Manta from Electrolabs and hear her summary on digital medicine, biomarkers, diagnostics, and therapeutics. Christine, over to you.
Hey everyone, thanks for inviting me here today. Glad to be here. So my talk focused on some foundational terminologies and guiding principles for navigating this digital medicine field. So my first take-home message was on the distinction between digital health, digital medicine, and digital therapeutics. What are we actually talking about when we throw those three terms out there? And the distinction between those three areas is based on requirements for clinical evidence and requirements for regulatory oversight. So when you're talking about something in the realm of digital health, there's no requirement for clinical evidence and there's no requirement for regulatory oversight there. As you move into digital medicine products, those do require a strong basis for clinical evidence. And they may or may not require regulatory oversight based on pursuing a medical device clearance. And then as you moved into digital therapeutics, those require both strong clinical evidence and regulatory oversight. Digital therapeutics are those that are delivering an intervention. So that's the key difference, evidence and regulatory requirements. The next definition that I talked about in my presentation was on the difference between a clinical outcome assessment and a biomarker. So a clinical outcome assessment, or a COA, is something that directly measures what matters most to people. And this is how they feel, function, or survive. And then a biomarker is a defined characteristic that measures a normal biological or pathogenic process. So if you're having a conversation with a patient or a study participant and they're telling you, I feel tired, I feel fatigued, I want to walk up the stairs without pain, that's them telling you something along the lines of a clinical outcome assessment if that's collected via a survey. Something like a lab test, that's a biomarker. That's giving you insight into that normal biological process or pathogenic process that's occurring. But a patient could have normal labs and still feel exhausted. So we want to understand both sides of that. And if you're talking about an eCOA, or electronic clinical outcome assessment, or you see the term digital biomarker, the digital or the electronic, that's just referring to the method of how that information was collected, whether it was collected digitally or via some type of digital survey or tool. And then the next foundational principle is that these digital clinical measures should be defined and deployed similarly across clinical research, clinical care, and public health. And this is a foundational principle that Electrolabs, along with DIME, Genentech, Myocardia, and some other groups put together into the playbook for digital clinical measures. This was launched in September of last year. And I'd encourage folks to definitely check that out to get some more details on how to do this well across those three areas. One of the key aspects of that playbook is to follow a checklist and the order of decision making in order to successfully implement remote monitoring across those 3 areas of research, care, and public health. And a foundation of that checklist, which is my 4th take-home message, is to follow the process of measures, technology, then operations. So first, define what you want to measure and why, what matters most to your patients or your population. Once you've got that decided, you can move into the technology. So what's the right tools for the job? Is it a wrist-worn product? Is it a mattress sensor? What do I need? What can I deploy that matches and has the measurement capabilities that I'm looking for? And then the last piece is operations. How do I get that technology out into my study to collect that data back? And by following measures tech ops, study teams, clinical care teams will avoid some common pain points and do this process successfully and get back meaningful data. Then my last take-home message is that patient engagement is required early and often to select a meaningful measurement process. I think historically, especially in the clinical trial space, we do maybe one focus group at the outset, get some information, and then move on with that. We really want to shift this model to engaging with that patient at multiple touch points throughout that clinical trial stage and doing regular check-ins to make sure that the outcome or the endpoint that we've selected tracks back to what we heard from them as being a meaningful measure that's going to drive a significant change in their quality of life. And looking forward to a discussion with the rest of the panel, and I'll hand it back over.
Thank you so much, Christine. And let's hear from our last speaker, Dr. Andrew Morris, who gave a great lecture on maintaining the integrity of a trustworthy health data ecosystem, and looking forward to hearing his 5 take-home messages.
So good morning, good evening, and I should say greetings from Scotland. It's actually our national day, it's Burns Night. So an action is I all want you to raise a glass of whiskey for us to support our economy. So it's a huge privilege. So I was invited to give a case study, and the hypothesis is on a nationwide basis, can you unite health data in all its multidimensional forms to enable discovery in a trustworthy way? So the case study was around Health Data Research UK, which is the National Institute for Health Data Science. And its aim is to unite, to improve, and to use health data on a population of up to 66 million people, the population of the UK. And my messages really relate to the challenges of creating and maintaining a trustworthy data ecosystem to enable that discovery, to enable that innovation at scale. So key messages are, what are the approaches to do this? And first of all, I think it requires collaboration, arguably on a scale we've never seen before. Firstly, we saw remarkable collaboration from funders. The Medical Research Council, Wellcome, British Heart Foundation, Innovate UK. In fact, now 12 funders have come together to support us nationwide, all aligned around joint strategy. So partnership, collaboration at scale. Second key point is around independence. So we are an independent body, a charity. So we don't sit in a university or a research institute, but we are neutral because we don't want to control data, but we want to enable its use at scale so that our analysts, my analysts tell me they spend 90% of their time accessing data, 10% of their time actually analyzing data. So how do we flip that equation so that in a trustworthy way, data are fair, findable, accessible, interoperable, and reusable? The third message is around trustworthiness, because one doesn't earn trust, one has to demonstrate trustworthiness in everything you do. So I'll be very interested in this discussion, especially Jennifer's contribution around how do we demonstrate trustworthiness. And some approaches that we found really useful is to engage the public in everything you do, especially in the design of the research, the infrastructure, the data sharing, and the services. So demonstrating trustworthiness with the publics and maintaining parity of importance of working with the publics is very important. And then my last message was around what are the skills needed to do this? And this is a flagship of two things. Firstly, team science, because we're working at the interface of mathematics, statistics, computational science, biomedicine, ethics, and the law. So how do we create porous institutions which actually bring these multiple disciplines together at scale? But most importantly, how do we do that to advance open science practice that's in the public interest for the benefit of mankind? And finally, I gave a case study of COVID, because certainly in the UK, what happened when COVID broke, the scientific community, in a very public-spirited way, all started to do their own thing. So there was an urgent need to do three things. Firstly, to coordinate the research effort. Secondly, to prioritize the research questions which were important to policymakers and system leaders. And lastly, to map, to flow, and to link the multidimensional data, whether it's genomic data, viral genomic data, clinical data, or even behavioral analytics data in a trustworthy way. And the case study I showed is how through that coordination and brokerage, we brought the academic industry, government, and clinical communities together. We report in a real-time basis. There's been over 1,100 research publications, either preprints or published papers, and over 240 projects are actually being supported based upon the national data infrastructure. And this approach has enabled things such as the recovery trial, which has recruited 32,000 people across 177 sites under the leadership of Martin Landry and Peter Horby, plus the COG UK, which is the viral genomics program led by Sharon Peacock, which has sequenced over 200,000 genomes. So in summary, I think we need to move from ecosystem to ecosystem, and we need to think of the whole if we're going to harness health data in a trustworthy way for the benefit of humankind. This isn't just a UK issue, it's an international challenge, and we're very privileged to be working with Gates, Wellcome, Minderoo to look at COVID-19 data sharing internationally, because only then will we actually derive insight that will combat the pandemic. So thank you very much, and I look forward to a great discussion on Burns Night.
Thank you so much, Dr. Morris. So I'd like to open the floor to discussion at this time and remind the audience members to please enter your questions into the chat feature, and we'll be sure to address those. And I'd like to pose the first question. We've heard a lot about trustworthiness, and I'd like to ask all of the speakers, what are some of the ways that we can instill trustworthiness and inform and educate patients about the essential trust issues in sharing their health data, why it's useful, but while also redefining the rights of patients as a safety issue. Jen Goldsack, I think you touched on that as your last point, and I think that's a great place to start. So how can we use existing frameworks, and what new rules do we need to put in place to redefine data rights as a patient safety issue, while also ensuring and educating patients and the public about their role in the process of sharing their data, and making these data rights something that's understood and important. So over to you.
Yeah, I'll kick this off, and I'm actually going to benefit greatly from going first, because I'm going to tease out some of the terrific points that my colleagues made around, we don't want to tell people all day that this is trustworthy. We need to show them. This is not a PR exercise. We actually have to demonstrate it. And so having a robust evidence base for everything we do is critically important, whether it's the tools capturing the data, whether it's the processes in place for protecting it and storing and handling it in line with how we are actually going to use it. That's critically important. The other piece that my colleagues teased out was around transparency. And the last piece, and then I'm actually going to answer your question around this idea of patient safety, is, I don't think this can, and I'm happy to be corrected, but I don't think that this sort of communication and education campaign is restricted only to patients as the audience. I actually think that a lot of the new potential risks, the new harms, the new considerations need to be communicated equally well to our clinical colleagues, whether they are practicing or whether they are researchers. We need to educate our policy makers on this. We really need to welcome, and this is something we try and do at DIME, and again, it's been a feature of all of my colleagues' comments, this is not a solo affair. We are not going to be able to do this as an army of one. We need to welcome our colleagues from other disciplinary backgrounds, whether that is sort of bioethics, whether it is sort of cybersecurity or whatever that looks like, so we can actually back up all of this with improved practices. And I'll make one last remark and then I'll throw it open to the floor here. There's this very nice term, and I've talked about redefining patient safety. And then for folks who may have watched my talk, I went to the WHO and I pulled their definition of patient safety, and it's absolutely lovely as written. But it talks about risks and harm, and it talks about based on the current understanding of what they may be. And I think there are new risks associated with these data, whether it's that harms can be derived from the data falling into other hands, all the sorts of risks and harms that Andrew spoke about, for example. The experts in analytics trying to answer pressing clinical questions are constrained from doing so because they are functioning as very, very overqualified administrators, right? There are new risks and there are new harms that we have to be aware of and that we have to educate on. And I don't think it's sufficient, although it's absolutely the flaw, that we educate our patient partners. We have to think about all of our colleagues at the table becoming more comfortable with these new concepts and their new colleagues and how to work with them to solve them collaboratively. Andrew, I don't know if you wanted to riff off that at all.
Shall I jump in and vehemently agree? So I'm not an expert in this area, but our colleague Sarah Cunningham-Burley conceptualized this in terms of how we engage patients, the public, practitioners, as well as your point. There's three ways. The easy way is to communicate what we're doing, but that's a one-way flow of information. Second way is co-design, to actually shape the research and the proportionate governance process. And thirdly is research to create evidence in terms of what works best. So we're very firmly in the co-design phase, and we embed publics throughout all our activities. A recent example, and I think this is important to demonstrate agility, what we wanted to do during COVID was to take a live feed of antigen testing positive patients and contact them to participate in a clinical trial in the community. And that is using routine data to actually enable the research process, convergence of care and research. And there are big information governance, confidentiality, privacy issues. So we mobilized a patient panel that we have anyway, and over 3 days, we got over 90 very detailed responses on how we should actually tailor that workflow of contact with patients. And now we're contacting 500 patients a day to participate in therapeutics trials in the community. Now, the message for me, I could not get 90 academics to respond to me in 3 days. So if you design your systems, it really does accelerate and enable the research process. And this is social science at the interface of biomedical science. We should do more of it.
And if I can ask a follow-up question to you, Dr. Morse, going off of your COVID example, we've seen that although this has been a very terrible time, this has also been a great opportunity to stretch the progress of, collaboration, education, public involvement, with a very rapid pace of discovery. I'm wondering, how can we keep this going in the absence of a pandemic, and how can we use what we've learned during, this COVID experience to establish some global best practices? So something that I think a lot of people feel is lacking is, in international collaboration, we don't have global best practices. How can we be sure that, regulations and data sharing are equitable, among these different collaborative parties? So how can we use what we've learned from this COVID experience moving forward in hopefully non-pandemic situations?
No, I think that's an excellent question. And actually, Steve Kern is speaking tomorrow from the Gates Foundation. And when he phoned me up in June, he said, I'm already preparing for the next pandemic. So, that's something that we should consider. The risk is we regress to the previous sclerotic mean of how we used to do business. So I think we are, I think COVID has been very enabling in terms of team science, collaboration, enabling regulation. But I think the challenge for us all is to ensure it leaves a legacy that is in the public interest. So ensuring that we perfectly describe the benefits of this new way of working collaboratively and how we attenuate and mitigate the risks around privacy, confidentiality is going to be very important. And creating a corpus of evidence which actually attenuates the risk of us actually just going back to business as usual and what I call death by committee.
Could I probably ask a question from the floor? Steve Kern, because you just mentioned him. Steve Kern just asked the question, I think patients first want to know what is a benefit to themselves, which is reasonable before they think about the commercial or the society benefit. So what would be your answer to that view? Because all the argumentation that we had so far was about risk, safety, and eventually commercialization possibilities. But if I have to give my data right now, as Christoph mentioned earlier, I would like to know what is my benefit directly now. And it's not yet there because it's not collected and it's not processed. So how do we get out of this vicious circle?
I'm happy to take a first cut at this, but I'll keep it brief so my colleagues can chime in. So I think two things here. First of all, I think let's not assume that patients are only in it for themselves. I think we should respect that we offer them something, but we should really ask them. And I think of the example of a patient with a rare disease, for example, they may be very happy to share that altruistically for the rest of their community, even if they may not receive an immediate benefit for themselves. I think in addition, we have to get better as a field, right? Whether we are talking about this particular topic, whether we are talking about vaccine missing, we have to do better as being scientific communicators. And so when we think about what is in it for a patient, for example, if we can establish a true learning healthcare system where information from an individual patient can inform where resources, where initiatives should be directed, because we're able to do that at a public health level, where we are able to improve systems and processes and develop new therapies and practices as a result of having good access and good visibility to these data sets. That is something that is very, very poorly communicated right now. So I don't want to dismiss the idea that we have to make the value proposition to patients. But I think that as we do that, we need to develop a whole new skill set as a field to be better communicators. I'm curious to hear what my colleagues think.
Maybe I can take that from here. I think, again, there are other areas of our social interaction that may serve as an example. Some of the very risky procedures. Let's take aviation. We see a clear difference between flight safety and, if you wish, security, and we understand the differences, and we compartmentalize the discussion in a useful way. So we want to make sure that, planes do not crash, and we want to make sure that planes do not get abducted or kidnapped or anything bad happens. And we have mechanisms, and there are times that we are discussing those mechanisms, and there are specializations across the society that take care of those mechanisms. If we then book a flight, we are not having a discussion on should I fly or am I assuming I'm going to crash. We have sort of separated that topic. And the reason for it is that we are looking at the statistics of, well, if I drive, how much more likely is it that I'm going to die if I take an airplane? Or if I take the train, how much less likely is it that I'm going to die if I take a car? And then we set that decision at some point, and we are no longer discussing this if we book travel. And I think, I think we need a clear-cut discussion between data security or, any of the adverse effect of the unintended use of data. We need to have a discussion, and we need to have specialists on trying to take care that that doesn't happen. But then the usage of the data and the analysis of the data should not be intermingled, with these processes. We really have to make sure that the public understands how many lives we sacrifice, how many hospitalizations we cause by not processing data. In fact, there are very few, if any, patients who have died from the loss of data so far, at least in our jurisdictions. Now, that may be dangerous in the future, that may be larger, but certainly we have a lot of patients who have to go to the hospital because the medication is wrongly dosed, is wrongly combined, because a problem, germs, go undetected, and other things like that, that by improving data processing may be much improved. So to cut a long story short, I think we need to have specialization and also compartmentalization of these discussions also in the public. And we have to get away from, even for simple clinical studies, intermixing the data security aspects with the actual patient safety / data safety aspect of what happens if I don't process that data. I find that very important, and even we as scientists and medical doctors are doing a very poor job, I should say, about informing our patients on what the danger of actually not doing something could be.
A practical example that comes to mind to me after hearing Jen and Christoph both speak is in terms of getting participants to enroll in clinical trials. Often a hesitancy that I've heard myself from acting as a clinical coordinator is, as a patient, I'm not going to get these results back. Like, how will I ever know how my data was used or impacted? Like, the results might be published, but will you notify me when that paper comes out? Probably not. So I think in terms of a practical way we can be more communicative, like Jen said, is to start sharing those results. Let patients know how their data is going to be used in the long term, whether that's months or years after their data has been used and the study is completed, so that they're truly engaged through the whole process.
And maybe just to echo my colleagues' comments, I'd be interested because I think what we've seen in the UK is what I call a thirst for science from the public that we've never seen before. There's a genuine interest in science. And I think part of the communication is to be honest. Healthcare is arguably the last major industry not to be really transformed by information. And the phrase I use is, it's at times like driving at night with no headlights if you don't have the information. So we need the information to make the best decisions for you. And finally, I think it is about being really crisp around how to communicate with the publics, because as scientists, we at times we're not the best communicators. But for example, in the current pandemic, we've used information to describe the risk in pregnant women or in children, these key things which people are interested in. And as vaccines are rolled out, people are asking, what is the effectiveness of this vaccine versus that vaccine? How long will I be immune for? So building a narrative and stories around how we use information, what the benefits are to patients and the public, and the safeguards, I think is part of this package.
All right, we have just about 1 minute left. If anyone has a final word they would like to put out there. Otherwise, I would have one final question, and that is the idea of investing and committing in team science as an idea and in young and emerging talent. From an operations perspective, what's the best way to go about this? You know, at the university scale, at the international scale, are there ways to implement this at a small scale and then establish some best practices to really ensure that we are developing ways of producing research and evaluating research that are sustainable and scalable?
I think the 1 minute answer is yes. We should absolutely be doing that. We need to take really seriously this idea of building an appropriately skilled workforce to be able to do all of the complex things that we discussed today, which is be able to do brilliant science, be able to appreciate some of the technical challenges that now we have to handle in a the digital era of health. And also to be able to do that critically important communication in a way that is palatable and that everyone can truly hear. I also would say that I'm not sure that focusing solely on training the next generation is going to solve the problem in the immediate future. I think we also need to find opportunities and ways to reskill and support folks who are already in the field. And, you know, that's something we try and do at DIME. And I know that I'm certain all of my colleagues on the line do that actively within their workplaces and within their communities. And I think that it needs to be a yes and approach, the current workforce plus a real commitment starting immediately to the future workforce.
All right, thank you so much. And in the interest of time, unfortunately, we'll have to end the session here. I'd like to thank all of the speakers for a wonderful discussion and for your lectures. I hope if any audience member has not had a chance yet to see their lectures, please do take the time to view those. They're really great. And at this time, we're going to be taking a 5-minute break before our next session, which will be moderated by Orla Smith, and that will be on how to judge translational progress and performance. So see everyone back here in about 5 minutes.
around, not ahead. A peak is just a way to walk at the sun. The forces on a hill, manifested by what you perceive as wind. Look around, not ahead. around, not ahead. The peak is just a way to walk at the sun. Look around, not ahead. A peak is just a way to walk at the sun. The forces on a hill, manifested by what you perceive as wind. Look around, not ahead.
Hello, I'm Orla Smith. I'm the editor of Science Translational Medicine. Welcome to our third session of Translate 2021, How to Judge Translational Progress and Performance. I would first like to introduce our co-moderator and then our three session speakers. So our co-moderator is Dr. Georges Schulz, who is Chair of Neurology at the University of Aachen, and he is also Vice Dean of the Medical Faculty and is a member of the permanent DFG Senate Commission on Clinical Research. We have three outstanding speakers for this session who will each say a few words about their own translational career trajectories, and then will introduce their summary slide of take-home messages from their video presentations. Our first speaker is Hans-Dieter Volk, who is head of the Institute of Medical Immunology at the Charité Medical University in Berlin. He is also the founding director of the Berlin-Brandenburg Center for Regenerative Therapies at the Berlin Institute of Health. Our next speaker is Hans-Gustav Ljunggrun, who is the founder of the Center for Infectious Medicine at the Karolinska Institutet in Stockholm, Sweden. He has served as Dean of Research at the Karolinska Institutet and is a member of the Nobel Assembly. And our final speaker is Jeffrey Bluestone, who is the president and CEO of Sonoma Biotherapeutics and is also the A.W. and Mary Margaret Clausen Distinguished Emeritus Professor in the Diabetes Center at the University of California, San Francisco. So without further ado, I would like to start with Hans-Dieter Volk.
Yeah, thank you, Orla. It's a big pleasure to be here. Hello, Jeff. I think my background is clinical immunologist, and I'm head of the Institute of Medical Immunology, as mentioned, and my special interest was for a long time development and of new biomarkers, new diagnostics. That was a focus for decades of life. But during the last more than 10 years, is more and more also new therapeutics, particularly in cell and gene therapy, which is a quite interesting way. And that was the reason that we founded about now 14 years ago, the BCRT, now the called BH Center for Regenerative Therapies. And the aim is clearly to develop or to bring things from bench to the bed. And we learned a lot, I think, in this process. How can we better motivate the people and see all the limitation. So today my talk is on translational measures, particularly the technology readiness level. Yeah, where's my slide? Okay, I have no slide, but I can do it for myself. And I think, yeah, thank you. I think to bring together the message of the talk is first, there's, I think, a clear consensus that the current system of measures and key performance indicators is not very useful for fostering translation like impact factor, citation index, age factor, and so on. And on the other hand, also the business models of academic tech transfer institutions, particularly also in the US, but also in Europe more and more, is they're looking for the break even to sell as much as possible for licensing. It's also not the best measure for translation, particularly for motivating the young people. On the other hand, we think measures are important for setting incentives of translation, but not the only limitation. It's also important for project selection, that we really train also the reviewers to look for the right criteria, and also for the monitoring of the projects by risk assessment and risk mitigation to adapt this. For us, the key success of translation is, of course, in our opinion, the impact on science and society is much more important than papers and patents and so on. But this might be applicable for the evaluation of translational centers, translational units, and so on, but maybe less valuable, or less suitable for checking of individual projects, individual teams, or even individual scientists regarding career track options and so on. I think if you define measures, it should reflect transparency, usefulness, feasibility, and consistency, which is, of course, not an easy tool. One tool which is used more and more, particularly in Europe, is the technology readiness level, which originally were developed by the NASA more than 20 years ago to bring from technology levels 1 to 9 to a commercialization. But it has been adapted by different ways, for example, organization in US like Beta and so on, but also in Europe. The European Commission used it now for more than 5 years in the Horizon Europe program, Horizon 2020 program. For any grant proposal for research innovation grants, you have to outline the technology readiness level, the aim, what you want, what is your starting point, what you want to reach. I think which is useful for discussion, for structuring consortia to discuss it, but have also many limitations because it's very rough and it's also not applicable to, not comparable for different clinical pipelines, for example, diagnostics or cell and gene therapy or repurposed use or whatever. And therefore we think there's a need for adoption. And that is what we developed during the last couple of years at our institution, to find some adoption, for example, for the cell and gene therapy, but also for enabling technology. This is very important to bring it to the way. And we formulated in the European Networks Restore, with one of the flagship initiatives for Restore by Health by Advanced Therapies, which try to figure out what could be good criteria. Yeah, I'm looking forward for the discussion. Thank you.
Thank you so much, Hans-Dieter. So now I'd like to hand off to our next speaker to introduce his take-home message slide, Hans-Gustav Ljunggren.
It's really exciting to be here, although not physically in Berlin. But I think the topic of the session here is really how to find metrics to judge efforts in translational research. And then thinking of this, I sort of got very challenged and by the title of the next talk of Jeff Blueston's talk. His talk was called Challenges of Rewarding Entrepreneurship in Translational Academic Research. And I was thinking along the very same line, but more in my case here on really challenges of rewarding enabling efforts of value for translational academic research. What's in my summary slide is really the description of the need for, or it's a description of successful translational research and really what it requires. And what it does require are really many efforts at many different levels, many levels which are maybe not immediately obvious or appreciated. One such example is, of course, long-term, it could be even lifelong organization and systematization of patient groups or patient data for that sake for future research, and or clinical sampling of a patient or patient material, patient tissue for future research. So in my talk I gave one example of a huge effort that we undertook, the entire lab almost here, more than 40 people, in establishing a large clinical COVID-19 biobank, which currently encompasses more than 20,000 frozen vials of human tissue, really dedicated for long-term future research. And because we think that a biobank like this could be of value, at least we thought it last spring, for a long period of time, 5 to 10 years. And it's, of course, when thinking about this, it's obvious that efforts like this require large resources. They require personnel deviating from regular work. And at the same time, they represent efforts that might not be considered to be of immediate value, and hence immediate merits are not collected, or because the value of it cannot even possibly be foreseen. So in engaging in such efforts, it's clearly so that simple academic metrics like impact factors, publications, and patients fall short. And the challenge, of course, which metrics also for efforts like this, that is essentially enabling efforts in translational research. So this is my little pitch, and I look forward towards a very good discussion.
Wonderful, thank you. And our third speaker is Jeffrey Bluestone.
Hi, everybody. It's great to be with you today, as even virtually, and I want to thank the organizers to really bring up what is really a continuing and I think really important area for thinking about how to move what are often outstanding discoveries in the laboratory into clinical and translational arenas, and ultimately drug development. In my talk, I gave a large overview because I think to be entrepreneurial and reward entrepreneurism, we really need to bolster the academic enterprise in a way that we just have not yet been able to do. I'm currently the president and CEO of Sonoma Biotherapeutics, but I've spent my whole career working at the bench in academic research, and really come to learn over those several decades in my pursuit of immune tolerance that it becomes increasingly challenging as the science gets more expensive, more complex, and how partnerships become more essential, whether that's partnerships with industry or government. And so the goal in my talk was to really emphasize that in order to have successful entrepreneurism, we need to build bridges that are at present still challenging to do. And so I left it with four major take-home messages for academic medical centers. First, our reward structure. We need to continue to evolve in being able to be supportive of cross-disciplinary research and education, and that first and foremost takes money. We can talk a little bit later, perhaps during a discussion, about the difference in the investments that are made in industry versus academia in terms of translational medicine, and how if academic medicine and its centers are going to really move forward, funding is critical. Infrastructure, I pointed out in my talk that the kind of costs associated with research today outstrip multiple-fold what it cost to do science 20 years ago before the genome sequencing and the need for these new tools. And investing in that infrastructure is going to be key. Obviously, academic promotion and tech transfer are part and parcel with being able to create a more effective environment for entrepreneurs. The second thing I pointed out is that team science is key. I've been fortunate enough over my career to be able to lead a number of international consortiums, from the Immune Tolerance Network to the Parker Institute for Cancer Immunotherapy. And what I found in those networks was the collaborative effort across multiple disciplines, which really led to better science and more creativity. It also allows us to work more closely with industry. Third, we need to be able to be more efficient in how we translate fundamental research, and having the ability to bring together a research powerhouse and a top-notch patient care center is really unique among academic medical centers, and that has to be exploited. Things like the biobanks we just heard about are key, but also how we work with patient samples and do research. And then finally, obviously, the educational and research approaches need to prepare for a diverse, global, and technically astute workforce. If we're not training that workforce in translational medicine, then we'll never be able to exploit that. So I look forward to the discussion and await my colleagues.
Thank you so much. So I have some questions that I'd like to ask the panel while we're waiting for questions to come in on the chat line. So my first question is for Jeffrey Bluestone. Why did you move from academia to industry, and how does this move relate to the challenges of entrepreneurship in the academic setting, specifically in the US?
Yeah, no, I think it's a question I'm asked a lot, having spent 40 years in academia. And I think there are a couple of reasons, which I think do touch on the discussion today. I've been working in translational medicine my whole career, having first developed a therapeutic, an anti-CD3 antibody back in 1987, and attempting to move that forward to develop it for patient treatments in autoimmune diseases like type 1 diabetes. And it's now 33 years later, and the drug is yet to be approved. Maybe this year we'll cross the finish line. And what I learned during that time, after going through 5 companies to try to do it, is that number one, if you really believe in translational medicine, you have to walk the walk. And too often, I think, founders of companies and investigators are relying on other people to do it. And I just felt for myself, for this next generation of therapeutic cell therapy that we have in the company, that if I was going to see this through, I need to do it. I can't be in the background complaining if other people don't get the job done. But I think equally important, I found that in the academic setting, that we didn't have the kinds of things I mentioned earlier on in place for me to be able to do it in academia. We didn't have the resources. I mean, I've raised $70 million so far for our company. I couldn't raise that kind of money in the academic setting. I was very concerned about students and their own independence and ability to be trained and do it. And a lot of success in translational medicine is team-oriented, and that wasn't being rewarded in my institution. And then finally, I felt that there was a need to be able to amplify the technology that we were going to be able to use. And that requires working with companies that have the resources to be able to invest in the kinds of clinical trials that we needed to do to test this drug. And I could do half a dozen phase 1 clinical trials, but to run efficacy trials was quite expensive. And so I think when I put it all together, it felt to me like if I was going to do it, I really better get into the company and see what I could do.
Thank you. I have a question for Hans-Christoph Ljunggren. How easy would it be for a country without a national health service to set up a COVID biobank like the one you described in your presentation?
It would probably be much more difficult, I would assume. I think it's sort of, I think one of the reasons why we did it was because we thought we could do it. We, of course, had to reorganize our whole lab. Everyone had to leave their job, their ongoing research to engage in this. But we could do it. And at the time, we thought there was a need for it. And so it's, of course, both about sort of the premises, the possibilities, but also the willingness to do something like this.
Okay, and then I have a question for Hans-Dieter Volk, and then we're going to move to George Schultz for the online questions. Hans-Dieter, what target should measures of translation be used for? For example, should they be used for evaluation of academic research at the individual or institutional level, or for grant approval?
Thanks, Ola, for this question. I think in principle, I would say all levels, because on one hand, of course, it's very important that we have measures which help to set incentives, which are not simply money or something like this. It's even more important to set career track options for young people to go in translation, because the paper output as the conventional measures are not as good. You have much less paper output and so on. And therefore, I think it's very important that this mindset is applied in such kind of translational ecosystem. On the other hand, of course, it needs also a training for the review process. That means if you really want to look for a good translational project, that means also the evaluation must be in the direction of medical need and of clear what do you want to reach, what are the milestones you want to reach, which is much more precise than in case of a basic science or fundamental research. And on the other hand, it's also very important to have the chance to fail, because that is, I think, very important, because failure, we can learn from failure, and it must be a culture that failure is not a bad thing. It must be published, it must be communicated, because we can learn from this. And this is also a kind of way that we need also the evaluation of processes, but also readjustment. For example, translation that is an advantage if you go from academic side together with spin-off companies and similar things, that you can come from the medical need and you can intensively look in the early clinical phase and can readjust the process to go back to the bench and back to the bed in an iterative way to improve this way, which is, I think, advantage in the classical conventional way if you go only from the industry way. And therefore, I think, in summary, I would say all levels are needed to support an ecosystem of translation.
Thank you. So we'll now go over to George Schultz for the questions on the chat.
Yes, we have received 2 questions so far. The first is by Ronan Rubinov, asking, one limitations of the field is that we don't teach translational medicine in medical school or PhD programs. How do we get these topics into the curricula? And that probably goes to all 3 of you, because you have different views in terms of your career stages. Can each of you give a short statement on that?
Yeah, I'd be happy to start. So when I was provost at UCSF, this issue came up very early on. In fact, at UCSF, being a research institution, there were lots of opportunities for medical students to enter into laboratories to do short or longer stays in research. And in fact, our MD PhD program was quite extensive. But the reverse wasn't happening. PhD students weren't getting experiences in clinical medicine that would help them think about their translation. And we did what we would call mini courses of 2 weeks, which included both didactic and hands-on experiences, where in particular graduate school courses, students would be able to work with clinicians, go into the clinics and meet and see patients up front, and be able to make real some of the book learning and lab research they had done. So I think going forward, it's going to be essential to have a kind of training, experiential training in both directions, so that PhDs can appreciate the value proposition of translational research by looking at those patients that benefit from it and seeing that disease in real time.
And you said that was a 2-week period, or?
Yeah, so we had 2-week mini courses that would happen several times a year. We had, we're on a quarter system, so 3 quarters a year. And so students were able to take that.
Yeah, maybe I can add something. Jeff, I completely agree with this. It was a similar thinking. And we founded, I think, more than 10 years ago, the graduate school, and exactly with two aims. One was, of course, to bring clinicians closer to the PhD work. That means we have implemented the clinician scientist work, which is a program, which means that really there is an off time of 50% of clinicians continue the clinical education, but also on the other hand, to work in the lab to learn the thinking of a typical PhD program. It's not an MD PhD program, it's really to keep the education in the clinic and in parallel, which is not an easy way, but it was very successful. Now it is used in the whole Charité, more than 100 clinician scientists, and even in whole Germany now, as a role model. The second way was the opposite, similar as you mentioned, the PhD students, to bring it closer to the bed, and that's exactly the same. We have each year 2 weeks courses that in our center, that PhD students, a young one in the first year and the second year, go to the clinical ward and look for the patients of interest, for example, in musculoskeletal system, transplantation, whatever, to get a better impression about what it means, what they are doing, to see the value of the research, which is, I think, very, very, very good incentive, much more than even money. And therefore, I completely agree that is a way we should even more intense for the future. And the third element is to bring together the teams, that 20% of our budget go to cross-disciplinary projects, translational projects, which are a mixture of PhD and MD, that they can cross-talk and learn from each other.
Okay, so I could give an example from the Karolinska, and it's really what's been pretty successful is to foster PhD students in a medical setting. So about 20 years ago, when I was fortunate to be able to fund a Center for Infectious Disease Research here, I actually asked to get it placed inside the university hospital rather than as a separate building next to. And many years later, more recently, when I had the position as the Dean of Research at Karolinska, a big process where lots of new buildings were being built, research buildings. We were moving from older buildings to new complexes. And we deliberately placed them on campus, very close to the university hospital, and linked essentially all buildings physically with sky bridges or similar to the hospital to get an integrated environment. And I think one physical proximity is one thing which is very important here, because it allows the spontaneous interactions between physician scientists or physicians and basic scientists. And it sort of creates a mental awareness that you are in a hospital and what you're doing is for a specific sake. So that's one example of activities.
Yes, thank you very much. Ola?
Okay, while we're waiting for more questions.
Oh,
to come
I
in on the...
have
you have?
I have more questions if you wish, yeah.
Oh, no, please go ahead.
Okay, then by Glenn Prestwich. Each of the speakers emphasized team science, cross-disciplinarity, science communication, passion together to clinic. How do patient advocacy groups fit in there? Who wants to answer that one?
We actually have a patient advocacy. We actually have such patient representatives in many of our in our steering groups, and it's of course key to have the patients with you, but it also creates a kind of awareness again among the scientists that what you're doing is sort of important and it's for real and it's for a specific sake. So.
Yeah,
sorry. I think I would like to add a similar way. I think it's very important. I have to say, we have here still space for improvement because we do a lot of interaction with the patient organization at the level of our research teams and so on, but less in the education. We should do it more. I think a good example is a rare disease. Here is a very active patient organization. But also very international. That is, I think, is a problem in Germany that many patient organizations, local organizations, don't speak in English, don't communicate in English, only in German. But our teams are interdisciplinary and also international. That means English is a language, and therefore sometimes not easy to find a way in the communication. But with the European level, is much easier because there's English the language, and therefore I think it's very important to have both the local one, the regional one, as well as the international level.
Okay, and then we go on. Yeah.
Yeah, just to add a last point, I've had a couple of very important examples in my career in this area. Obviously, in the HIV community, there was a tremendous amount of advocacy, as well as in the stem cell community of disease when we were passing Prop 71 in California. And what I found the most important elements, besides the kind of input of patient advocacy groups, was the passion that got created by bringing people in for seminars to talk about their disease, to talk about the field. And so I think we can be involved with patient advocacy groups, not just to be involved in leadership and steering committees. That's important. But as the passionate few who can come in and really help the students appreciate the importance and value of the work that's being done in translational medicine. And I have seen students come out of talks by individuals, kids with type 1 diabetes, adults with cancer in the cancer immunotherapy space, and seen a transformation in their commitment to the science after hearing from these patients.
If we move on to the next question, Ola, you just interrupt me if you have a question, please.
Okay.
So what is the current model of formal education versus mentorship and apprenticeship in translation? Which is more important for long-term impact? So is it formal education or mentorship?
So I don't see this as an either or. I think we need to have the mixture. I think what Hans-Dieter talked about, which is those first couple of years when a student is gaining their feet in science, having more formal education, especially interacting with clinical groups and like, I think is really important. But at the end of the day, of course, direct mentorship and how the mentor helps the students and the postdocs understand the value of translational medicine, brings them to team meetings, incorporates the students in the translational enterprise. That really builds a solid scaffold for those students to go out and do it themselves. So for me, I think it's a balancing act. You don't want to have too much of one or the other, but putting it together is what works best.
I would think in addition to that, sort of the mindset among the leaders, among the mentors, among the group leaders, among the unit heads, the department heads is important. If you drive towards translation as a leader, people will follow you. And if you drive against, that will not promote translation.
I see it similar. I see it's not an either or, but as a mentorship is a very important aspect, not only for, as mentioned before, for that people follow you. I think it's also important to select the talents, the most important talents. That is by both sides, PhDs or MDs. And therefore, I think that is not possible or much more difficult possible in a formal system only. You need mentorship, and you have to find the talents which are, need direct support and not the normal career pathway to accelerate it and to use them to use the capacities they have. And therefore, I think it's very, very important to have both access, the direct mentorship as well as the program.
I think the next question is a very important one. It relates to when should translational research be considered by a basic scientist? It reads, should more basic research be approached with an eye to the next translational stage? Perhaps the regulatory steps should be considered within basic research.
Maybe I start. I would say it's similar as before. It's not either or. I think basic science is very important, but in this way, normal definition of basic science that is undirected. You look what you can find and what you can explain, and that is very important. And the best things can go in translation, but it's not automatically the aim, and it shouldn't be the aim. But on the other hand, I think I would say a mission-driven basic science. That means a science which is a basic science which is following a mission, for example, to bring a new gene therapy or whatever. Here you need really basic scientists to improve it and to come to a new level of understanding of failure and allowing refined translation as mentioned before. Therefore, I think mission-driven science and the mission you want to get is a very important point.
One of the things we have to be careful of is that we don't throw the baby out with the bathwater here, as they say in the US. We need to continue to respect, promote, fund, and emphasize basic research. It's absolutely essential. It's the cornerstone of academic efforts, and it really is really the food for translational research. So we can't go overboard one way or the other. So I feel like the goal here is to provide opportunities, to give the students and other young faculty opportunities. And in that regard, opportunities in learning about regulatory sciences are important as well, because one of the biggest challenges, especially if you're not a power user, is to move through the regulatory system. But I would point out there that it's also best, I think it's best accomplished by having academic medical centers devote some resources to building regulatory units that can help facilitate the research, rather than asking the young faculty member to do it all themselves.
Absolutely, I completely agree. I think we need both. But on the other hand, there exist also translational projects which need some feed from basic scientists if they are interested on this. And somebody is interested to do it at least for a time, to bring their own project to the translation. Others say, okay, that's not my job. I bring it just to the first level and then I go back to basic science. I think both is possible. We should not dogmatic. We should be here open and give opportunities. And that, I think, exactly the opportunity or the advantage of a big center.
I mean, I agree with all what you said. I mean, we should, of course, maintain basic research. That's very important. But once you discover something that could be translated, there should be the possibility to translate it. If that possibility does not exist, it's not good. And then it's not so obvious all the time that someone else will translate it. I mean, I think many people have the experience that if you have discovered something, you need to translate it yourself, maybe until the value of it is seen, and then others pick up on it.
I think we just have time for one more question. Is there one more question in the feed, George?
I have exactly one more. Yes, that's
Okay.
correct. And then you wrap up.
Yes.
And the question is by Ali Mobasheri. It's translation and animal models are important. How can we facilitate the development of better translational models for difficult to treat diseases?
I think Ali's question is really an important one, and one in which one needs to be spending a lot more thinking and time. Number one, and I mentioned it in my talk, that industry is very reluctant sometimes to collaborate and partner with academic medical centers because the data suggests that a number of the discoveries are not repeatable. The second problem is that there's a mantra out there that the animal models do not represent humans. Now, we are very much improved in our ability to do research directly with human material, and I think that's really important. But for a number of aspects of precision medicine, when you're going after common pathways of disease, the animal models are essential. And so I think what we need to do, again, is to combine and do reverse translation. We always think about translation in the forward direction, from basic science to medicine, but we need reverse translation as well, where the patient material and the disease itself is studied to look for similarities as we develop our animal models so that they more closely reflect the human disease. And so I think, again, here's where partnerships, team science, and collaborative efforts really can make a difference.
I could maybe add, it's not on the animal models, but I think something that is a bit underused maybe is, assume we do a clinical trial in humans, that is the possibility to use that patient for exploratory analysis. For example, when we carry out cell-mediated immunotherapies, let's say in a clinical setting, to really try to explore as much as possible what we do and try to learn as much as possible. That is not really a direct answer to the question. I agree with what Jeff said, but we shall not underuse the potential that we have created. Of course, it has to be done with the proper ethical permits and regulatory permits, but there is quite much potential there.
I completely agree in this way. That is, I think, a very important point that we can learn a lot from early clinical trial to de-risk late clinical development, because I think the problem is the most failure in clinical trials we have in phase 2b and even in phase 3 because of efficacy and not in phase 1 by safety. And therefore, it's very important to learn as much as possible in the phase 1 to A about the mode of action, about responder, non-responder, and so on. And I think there's a rethinking also in the pharma industry. Because we do the job now for 20 years to monitor clinical trials, and we have also a spin-off company founded and so on. And what we learned is that there's a completely change. Today, in the last 2 years, for example, we are involved in many clinical trials of completely new drugs, and we can say the industry invests twice more for the biomarker than for the clinical trial by itself. So it means more and more learning from the early patients as much as possible to design your later clinical development, and therefore it's very important. And you can also use it to go back in a reverse translation to adopt your animal models, for example, with aged immune system, other things.
Great, thank you very much. Unfortunately, I think we're out of time. So on behalf of George Schultz and myself, I'd like to thank our 3 great speakers, the great questions from the audience. I believe we now have a 10-minute break before reconvening for the report on consensus process 1, which will be chaired by Glenn Prestwich and Hans-Dieter Vogt. So thank you very much, everyone.
Thank you. It was a pleasure.
Thank you.
around, not ahead. The peak is just a way to walk at the sun. Look around, not ahead. A peak is just a way to walk at the sun. The forces on a hill, manifested by what you perceive as wind. around, not ahead. The peak is just a way to walk at the sun. Look around, not ahead. A peak is just a way to walk at the sun. The forces on a hill, manifested by what you perceive as wind. Look around, not ahead. Look around, not ahead. The peak is just a way to walk at the sun. Have nowhere to look. Look around, not ahead. A peak is just a way to walk at the sun. The forces on a hill, manifested by what you perceive as wind. Look around, not ahead.
Ich bin nur für die Questions. Hm? Good. Welcome back. Welcome back to the Translate event. We're now talking about the consensus papers. There are 2 consensus papers. We invite you, if you haven't done so, to comment on those on the web page using the email buttons under the consensus papers. With that, I'd like to hand over to Glenn Prestwich to explain us the history of the first consensus process. Glenn, can you hear me?
Yes, yes. Am I online?
Yes, you're online. If you could explain how we came to the current version of the consensus paper together with Brenna and Hans-Dieter.
All right. We chose this topic of how to measure translational impact based on prior efforts to discuss translational research and how to get to the end. But now, how do you measure it? How do you measure the impact that you're making, as well as the impact on the careers of the individuals, the impact on society, and the impact on individual patients? So in an effort to try and make this understandable or kind of reduce it to something that we could measure, which is always a challenge, we came up with a number of ideas that are outlined in the Measures of Translational Impact consensus paper draft, which is posted online and is what Georg was mentioning, that we should elicit email contributions on views, suggestions, and responses to some of the things that we clearly left open. It's a very rough draft developed by Georg and David Granger and Bob Goldberg, myself, Brenna Rao, and Hans-Dieter. And so why don't I take this opportunity to say a quick introduction for myself. I'm involved with translational research, have been for some 20 years, both as an entrepreneur and a faculty member. And I continue that here in Spokane with creating a new incubator space. And Brenna, if you just want to do a quick introduction of yourself, then we can proceed from there.
Sure. I'm Brenna Rao. I'm in Portland, Oregon. I'm managing director for Bioscience Incubator here as well. But prior to running 12 Bridge, I oversaw technology commercialization and technology transfer for multiple universities, including UCLA and OHSU.
So we bring a multi-university approach, both from the faculty side and from the entrepreneur side, and in Brenna's case, the investor side, to try and understand what it is that is important to the people generating the research and trying to translate it out of the academic environment, and to the other side, which we've heard a lot about, which is the hospitals and the patients bringing it forward towards the clinic. So maybe we should talk about this. Are we going to be having question and answer on this, or is this more or less a discussion among the two of us, well, three of us? I think Hans-Dieter was going to join us.
Hans-Dieter is also here, and I have in front of me the question feed. If people type in their questions, they can reach us, and I would then, once I get questions, feed them in. But I think it would be good if you briefly explain how we came to the current version of the Translate Consensus Paper 1, maybe also from Hans-Dieter's perspective.
Yeah, okay, thank you. I think Glenn have wonderfully introduced it, how we started and how we discussed in the last couple of weeks in virtual meetings and by email and online documents. And of course, it's only a draft. It's only some ideas. It's not finally structured and finalized, and therefore we need your input. I think it's very important as much as possible. And I think as mentioned before, I think there are two key messages. The first one is absolute agreement that the current concepts or current measures to make analysis of translation research are not sufficient, and therefore is a need for other measures. But the most important one is really what is a key aim, and the key aim is clearly to have an impact on the society and on patients and on science. That I think is a very important incentive also that the people understand they do something meaningful. They give something back to the taxpayer. They pay a lot of money for our research. And this looks very theoretically, but I think it's a big, important thought which is accepted or taken by many scientists, young scientists, because they like to do some meaningful. Money is not the half story, but of course it's also an important point for the career science. And therefore we structured in a different way what is not working we think, what could be important, like I mentioned before, and what kind of measures we have for individual and individual teams and individual people, career track options to measure. That was a principal structure, and we tried to bring it together, and we would be happy to get feedback.
And one of the ways in which we were going to encourage feedback was with creating a graphic. And the graphic, if you have a look at the uploaded or downloaded, in your case, draft, you'll see figure 1, a multifactorial landscape. The notion here is that if we're measuring translational efficacy, as it were, we're not measuring a single parameter. We're measuring a lot of parameters. And in fact, 3 parameters, 3 axes for this landscape is really not enough, but it's hard to draw 4 or 5 dimensional structures in a journal article. So what I suggested here is at least 3 things that were important: is the number of people affected by, impacted or affected by a translational bit of progress in the clinic, the effect on society as another axis, how not the individuals, but how does society as a whole benefit, and what is the individual benefit for a specific bit of technology. So you can imagine curing 100 kids in a rare disease of cancer saves their lives. That's an enormous impact that has important societal benefit, but the major beneficiaries are the kids who survive and their families. On the other hand, you have things like drugs to treat headaches. Billions of people are going to use those. It's making a marginal effect, but it's an important cumulative effect of cumulative translational impact. And then the number of individuals affected with something like essentially a cure for type 1 diabetes would be in millions, tens of millions of people. Also a huge societal impact, both on families, individuals, as well as economics for that magnitude of people affected for their entire lifetimes. Those are not the only 3 axes. And so other things that we've suggested are things that could be return on investment, or scalability, or disability adjusted life years. And in fact, one of the ways that we decided to come to this suggestion was by talking with experts, with voices from the real world. And so another part of this paper this consensus paper will include boxes, if you will, voices from the real world, we call it, to have a few people who are actively involved in translational medicine, who are not authors or not someplace else represented in the symposium, have them speak about what they feel the most important thing is in terms of what you can measure and what the important outcomes are in terms of translation. So that's another feature. So I'll be quiet for a second.
Just, Glenn, I just received an email or a message, a question from Jochen Büchel to Brenda. In Portland, is there collaboration with cultural studies? I understand that question a little bit. Is that focusing broader than just on the translation itself? Brenda, would you like to comment on that? I hope I could explain the question good enough.
Could you restate the question, please?
In Portland, is there collaboration with cultural studies, is what Jochen Büchel asks. So how are sort of other elements, so that's how I read it, other social elements, social science elements included into considerations?
I think multi-factor consideration of cultural elements and multi-factors are relevant for translation in any context, certainly in Portland. I mean, within Portland, the socioeconomic context of the patient populations that are served here, the types of research that are undertaken here, the dynamics of the researchers, all of those factors play into the efficiency of translation. So I think the cultural context is relevant. I think one of the key challenges for an institution like OHSU is, as an academic medical center, it doesn't necessarily have the holistic perspective of all of the fields and domains to bring the full cultural perspective. So looking to external stakeholders to bring in those inputs into the process.
Another question, I'm just...
Maybe I can add
oh,
something?
Please,
Yeah,
please go ahead.
Maybe I can add something. I think we had a big discussion in the framework of the Restore flagship initiative here in Europe. And I think there was also one conclusion is that particularly the ethical science, the ethics is a very important point to bring in this system, which is sometimes a little bit, we see it only, we need the ethical approval for our clinical trial or whatever. That's not the whole story. Ethics much more. And we see it particularly in this modern world, for example, that means also availability and affordability of new drugs, new treatments, which is a big issue. And we see it, how much discussion was, for example, with modern gene therapy and rare diseases, that you can cure patients, but it's not available for all. It's not only by the pricing, it's also by the number of sources. You see it now with the vaccine for COVID. We have fantastic development. On the other hand, the availability is quite low. That means it's still very limited, and there is a big discussion, is it right, how we selected the order of vaccination? And since such kind of ethical aspects are also very important for translation. But it was not so much the focus of this paper, because here was a question really for measures. But maybe we have to think about how we bring in such elements as well.
Just if I may, I would also ask the next question from Ulf Tölch. What about the robustness of the underlying research? Is this not, or is this the sine qua non for all our excellence in science and for all other measures that are derived from that? Which is a little bit an overlap to the other consensus paper, but I think it's a fair question to bring that up here. Anybody from you three who would like to ask a reply to that?
Well, I think one of the, I'll just make a short comment. I know that industry is very concerned with the low level of relative reproducibility of academic data. The academic data is generated in order to achieve academic excellence and progress through either a degree program or the tenure ranks. And so that's always a built-in conflict of interest, if you will. And that data reproducibility, I think, is essential to address, so that part of the metrics of translational success at an academic side should be reproducibility of data. It needs to become data, not just authenticity, but data reproducibility. It's always been passé to go and do somebody else's experiment in your own lab. How do you publish that? Maybe that is an ethic that we need to change.
But would you see that as a part of this paper, or is that rather in the other paper? It's a little bit the question here. It's about metrics and measures. So it's a pre-requirement to measure at all, to some degree.
Yeah.
I think measurements of translation very much depend on where you sit within the ecosystem. When we think about innovation, we often talk about it as though it's a linear process. But in the real world, innovation is complex, multifactorial. It is full of dead ends and unexpected consequences. So you need to take a holistic perspective. And I think that's part of what this paper is trying to get to, is that there are many metrics that we can look to. And for a given stakeholder, for a given perspective, a different set of metrics might be most relevant. So when you're talking about an individual researcher in the lab versus the goal of an academic institution, you may key on different translational metrics. So I think certainly the integrity of the science has to be the starting point for all of that. But I think in looking at this question, we share the view that translation is not, the mission of the science is not done until translation is complete.
Probably one last question that I have here in the feed is from Hans Lemus. He asks, in case of figure 1, I think the question goes to Glenn, in case of figure 1, who will be responsible for measuring these indicators in order to guarantee a fair and objective comparison between different projects? So it's nice to have a measure, but who's then in charge? I think that's a very valid question.
Absolutely. Therein lies the rub, to quote Shakespeare. That's really the problem, is what do you measure and how do you measure it, and how do you how do you validate your measurement tools? I don't have that answer. That's why some of the things that we suggested, for example, Maurizio Vecchione, who's one of our in Voices from the Real World, his bottom line is that he wants scientific research and innovation with impact to produce a double bottom line of investor return and social impact. Now we're way at the other end of what's going on with translation. And that's the other end of, how many papers did you publish and how many patents are there and how many companies did you start and what's the dollar value, what's the dollar cap of the companies that you started. So there's a huge gap in the middle. We don't know how to measure it. The easy things to measure are not necessarily the ones that are the leading indicators that are going to tell you success, predict success. That's the problem, which is rephrasing what Brenna said in different words, I think.
Okay, thank you very much for that discussion and for presenting this consensus paper 1. And now we would like to proceed to consensus paper 2. And I would like to invite Petra Reinke to me here on stage. And together with Ronan Rubinov and Uli Dirnagel, we are going to talk about the proposed consensus paper 2. And thank Brenna and Glenn for their participation here and Dieter on stage. And while we have the switch, we are already there, perfect. Then I think, Ronan, would it be okay if you could briefly introduce to the consensus paper 2 and then Uli Dirnagel joins in and Petra, and we make first a presentation of the current status of the paper and then go further.
Certainly. Can you hear me, Gary?
Yes.
Okay, so thank you. This is consensus paper 2, and you have the authors here. It's Ulrich Dirnagel, Georg David Granger, Petra Reinke, and myself. We really focused here on the role of data reproducibility. The challenge has already been alluded earlier, the issue of how many findings fail to reproduce even in the animal experiment or preclinical experiment. And then the additional concern of how many of these fail to translate when moved from other models to humans. Now, some of that is inevitable biology and the difference in species, but there are also other issues that we want to challenge here as to hopefully give a framework where we could try and reduce some of this variability and try to improve the throughput so that we have a more robust system. And this includes thinking about robustness and reproducibility, thinking about generalizability from one lab to another, making sure that experiments are done in a robust way that has statistical power, that is clear on the methods used, and that allows other labs to easily and rapidly test the original finding. And then the translatability of preclinical models. In industry where I work, we try to get out of preclinical systems and into humans as rapidly as we can. And we don't really want to spend a lot of time on that last 1 or 2% of improvement in the model. In other words, to make sure we get what we need, get fit for purpose information, and get sufficient data to try and test the hypothesis in people. And I think that's what this consensus paper is about. As for paper 1, this is a rough draft, and we really appreciate people taking some time to read it and give us feedback. Let me stop there.
And I should have properly introduced Ronan as he is in charge of R&D in Novartis and helped in this perspective with the paper. And Uli Dirnagel, he runs the Quest Center in the Berlin Institute of Health, where the focus is indeed on increasing quality of preclinical data. And Uli, would you like to add from that perspective?
Well, I'm also a stroke researcher, and so I've come from a field. Yeah, I'm just mentioning it because this is a field that has particularly hit hard by translational attrition. In fact, we have spent decades investigating mouse and rat models and are very successful in treating those model diseases. But basically none of this has made it into clinical practice. So we have produced a lot of fancy papers, but very little benefit for our patients. So that's for me, at least, that's a very strong incentive to think about those things. And I strongly believe, and this was also a theme in the paper, in the discussion before about the consensus paper 1, I think the basis for all this translation, whatever it is, must be the robustness of our data. And so I think that's, to me, this is absolutely key. And I have good news, I think, and I think we have also put it into this draft. The good news is that unlike many other issues of translational problems, here with those problems that have to do with the quality of the data, we know what to do. So basically, it's amenable. We can improve on it. Meta research and reproducibility projects, I think, have shown that there is a problem and where the problem is and how we can alleviate it. And so this is about increasing power. It's about increasing in the internal validity, reducing bias, increasing external validity. I mean, so the good news here really is we know where the problems are and we can work on them. And as in the previous session was already mentioned, I think we need to incentivize this type of work because at least in the academic current stream, it's about producing fancy papers and then it's kind of hit and run. Then you go to the next paper. So I think we can improve quite a lot.
And I'm in the luxury situation to be together here with Petra Reinke, who is in charge of the BCAT initiative and building that we are generating here in Berlin for cell and gene therapy. And she is together with me in the BCRT, active in bringing regenerative medicine forward. From your perspective, Petra, what is really important in this topic, in this dimension that has probably not been mentioned so far?
Also many things are already explained. I would say, because I'm a clinician and I have seen at any time in my life a patient in different clinical situations. And the point is, if you want to translate basic research, you have to find a proper mindset, and and the mindset is, let's say, related to the opportunities to give to the researchers and also to the clinicians. On one hand, it is related to the knowledge, and the knowledge doesn't mean only the basic research knowledge. That is a fundament for all of us, but you have to know also the knowledge with respect to the regulatory framework you have to fulfill at the end if you want to go to a phase 1 to a first-in-human clinical trial. And this is sometimes, or not even, unfortunately sometimes, this is in many of our clinical situations in which we speak about translation missing, that this knowledge which impact our animal work or our so-called basic research is not well accepted or well known, and therefore you start at several time points, even with your basic research, from the scratch. This is one point I want to mention, and the other point is related to the animal work and related to any kind of basic research. We have, I think, to accept in the community that in biology we have to face diversity. It is not a matter that you have one experiment which relates to one result. You have at any time so-called outliers which are mostly ignored due to several reasons. And one reason is that we streamline our data, we streamline our hypothesis. And this is not fitting to the real patient world. And this is also one point we have to keep in mind if we speak about translation and translatability of our data and filling the gap between basic research and the phase 1 to A to B and industry collaboration.
Thank you. We have a question from Steve Kern. GXP, so GMP, GLP, GCP, offers straightforward ways to standardize experiments so that they have a chance of being reproducible. Yet academics tend to cave at its use. They see GXP as bureaucratic rather than quality adding. How do we tackle this and create a culture that views reproducibility as a quality? That just here into the discussion. It's a little bit linking what Petra just said to Uli Dirnagel.
So maybe I can answer something here. Actual GXP is really quite expensive because of all of the, essentially bureaucratic, but all of the tracking that's required to create a chain of evidence for regulators. But we can do GXP-like experiments in academia to make sure that we capture the information and that the publications actually allow the kind of tracking that's necessary for reproducibility. That's actually a pretty easy fix, and I think that's one we should encourage.
Maybe I can just add to that there are several initiatives. One, for example, by IMI, EU funding, it's called EQUIP, and we have a local one that's called Premier. And these are initiatives that develop, as you just said, kind of light versions of GLP. So these are quality management systems that are amenable also to academic laboratories. So I think it is possible, but we need two different things in addition to what we have now. One is we need additional resources because even if it's light GLP or GXP, it's resource intense. And the other, and that's probably more important, it also involves a culture change because this type of quality needs to be incentivized. So in promotion, in tenure and so forth, we have to look at those things. As long as we don't do this, why should an academician do that? I mean, we get tenure for a fancy paper, and you don't get fancy paper for this type of quality. So we need a culture change here, I think.
I think that's a very important point that you raise, Uli, that it's also in terms of the incentives and essential how we drive those systems. From what I've heard, also the variability, what Petra raised, the diversity that we have in the patient population, bringing that early into the equation. Is that something that works with the classical GXP structures in preclinical ways, or is it rather the motivation to go faster into clinical trials under controlled conditions, etc., but motivate rather that faster track towards reality check? That's how I understood Ronan a little bit earlier, and also Petra. Would you like to comment on that? So motivate people to jump earlier into the cold water.
I would strongly recommend to go faster in the cold water, to put your words again on the table, because all our animal work, even if it is of importance, is not really fitting to the patient world. And therefore, I think we have to have, of course, a good explanation for our hypothesis. That means we have to have as much as possible an understanding about the basic mechanisms we want to tackle. But then we have to move very fast and under ethical conditions, of course, to the first in human, to figure out safety issues, get evidence or some hints for effectiveness of our treatment strategies, and even to figure out which of the patient population do not fitting to this kind of strategy. We will not have success with any therapeutic applications in 100% of the targeted disease.
Now I have a nasty question from my perspective. You are focusing on cell and gene therapy. Ronan does drugs, and Uli also is focusing on drug therapies. I'm coming from a domain where medtech devices are relevant, and we have learned over the last decades the very hard way, I mean really hard way, that it's not only about the product, but it's the way how it is brought to the patient, so how the therapy is realized. So in that sense, the surgeon, for example, in joint replacement surgery, for example, plays a highly relevant role in this whole equation. So you may have a perfect solution, you may have the perfect product, the perfect therapy, but we say that sometimes we need to be dump-proof, so it needs to be working even in somebody who has no understanding about it. How do we solve that aspect of robustness and reproducibility in therapies, sort of beyond the classical pharma and classical ATMP domain, where we urgently need robustness, and that's even the motivation for a whole new regulation world, the MDR, which is not yet feeling that it's tackling all those risks appropriately, probably. Any comments from your side?
Yeah, it's a nasty question. That's right. I think it's a relevant one and a very hard one to answer. Look, the whole issue is scalability, right? I mean, the old joke that if exercise were a pill, everyone would take it applies. And in fact, one of the big challenges for us is how do we create, as someone said in an earlier session, drugs for headache and hypertension for 1 million people and a bespoke gene therapy for 10 people. And it's a real completely different game at each end of that scale. And I think you're alluding to this as well. We will, I think, first have to demonstrate that the mechanisms that we're testing and proposing are robust. And if that does work, then I maybe optimistically think that a commercial model can follow. And there will be a company that will take it forward, or an academic partnership with patient advocacy groups and biotechs to make these things available. And I think those bars will get lower with repetition. And I think we have to keep at this, because at the end of the day, all of this is a game for optimists, and we really need to keep that in mind.
I like that. Uli, any remarks from your side?
Well, what you just described, I think sometimes called translation 2, sometimes translation 4. But for me, still, these further steps of translation, they still only make sense if they build on something that is robust and reliable. So we should take it step by step. So if translation 1 is not working, you can't do translation 2. So, and by the way, this problem that you just described for medicinal products exists for drugs just like it. I mean, you can have a beautiful pill for hypertension, but if people don't take it, or there's a problem in taking it, it's of no use. But I think this is a whole different issue. It's problematic, and we need to take care of it. But it has nothing to do with what we were talking earlier on, which I thought was the transition from modeling to first in man. And I think there's one very contentious issue here in this discussion, which we did not have and still don't have the time to discuss about. And that's what Petra says. She basically says, well, we need a little bit of, I'm exaggerating, you need a little bit of basic research to get some idea about the phenomenon, and then snap, you take it to the patient. Don't waste too much time on models, because models are not worth it, they are not predictive. And I'm ready to buy into that. For some diseases, for some others not. And we don't know which one is which. And my last point here would be still the current model. This is not the current model. The current model, at least our argument is always that we are going to go for external validity, for construct validity. And then if we have something and it's robust, and then we take it to humans. I think this has still a lot of value. It hasn't been very successful in many cases, but the model, I think, is not dead yet. And in fact, what Peter said, well, let's be fast, let's go fast into human, but ethical. But this "but ethical" is a bit of a problem here, right? I mean, we need to be aware of what we are talking about here. This whole part in the animal is to be ethical about it. So I think we still have a lot of room for discussion here.
And I would like to give a little the chance, Petra, to reply to your points. And Petra?
No. I wouldn't reply to the points, because then the discussion will need longer time. So we can continue this on another time. I would only add one point with relation to your question. And this is that at least for cell and gene therapy, you have to have already from the beginning on in mind the technology and the process you want to use for your cell and gene product development, which means you have to have at least three aspects. One is the basic research, one is the technology development, which is likelihood to achieve any kind of standard which then can taken over by industry that is not related to a full upscaling strategy, and you have to have at least enough data that the phase 1-A is so-called successful with relation to safety and hints for efficacy. This is what I have to add from my point. Yeah?
Thank you very much. Thank you very much, all three of you. And we are now processing to the next point in the program. And I would like to invite now Klaus Nitschke. But before coming to that, I would ask you all to use the Challenge at Translate button on the homepage. We have been able to select 3 pitches of translational stories. We will see if they are successful. They are in a relatively early phase and tell the story of their translation endeavor, and have a look at those 3 stories. We will ask you to vote. Which one do you like most? Click it on the homepage, have a look at those, and give us your opinion. We will have, after the introduction to the digital labs by Klaus Nitschke, the chance to listen to those pitches and see and talk to those who are representing the pitches. Now, the floor is yours, Klaus Nitschke, and I'm very happy to see you. You're unfortunately not here in Berlin, you're in Bonn, and we have the tight link and are looking forward to your introduction to the digital labs. Klaus Nitschke is at the Berlin Institute of Health, and he built, we're talking about translation, and he built, if I want to be a little bit nasty, a translational machinery. So a machinery that produces products like companies. And we'd like to hear a little bit more about that role model or what you built there and how successful it got off the grounds. So the floor is yours.
Thank you, Gail, very much for having me on this Congress.
I'm very happy to be here, and I'd like to introduce to you Digital Labs. And just a second, I open up a presentation, and hopefully this can be seen. So Digital Labs is a unique translation engine for digital health in research hospitals. Digital Labs belongs and is tightly linked to the technology transfer unit of BIH. The technology transfer unit is called Berlin BIH Innovations. Within BIH Innovations, Digital Labs is one out of 4 units. And the reason why we created Digital Labs a couple of years ago was to drive translation in digital health. And we are all aware that there is not enough translation in digital health in research hospitals, especially in Germany. There is many assets, sufficient ideas from all the marvelous clinical researchers. We have abundant and increasing data within the hospital and within basically research projects. And we see that there is technology available and it is affordable. Just for example, sensors. Sensors decreased in price over the last couple of years. Let's say in 2004, one sensor was $1.30, and now in 2020 it was 38 cents. In addition, there is nobody to execute. So due to the fact that there are researchers and that there are MDs, there is a lack of skill with regards to how to build digital translational products. There is no digital mindset to develop digital solutions. And as a result, we don't have enough digital products and digital solutions to improve patient outcome. At the same time, we have a huge potential, especially at Charité, which needs to be leveraged for translation. So just some examples. We have 3.5 million image data. So it's CT, MRT, X-ray. There is 27 million lab data. Marvelous clinicians, researchers. So at Charité, there are more than 4,000. We have know-how within BIH with regards to how to innovate. We know how to build certified and validated digital products, and we know how to market them. In addition, there is trusted brands for most Charité. All this basically made us to think about, okay, how to improve the current situation and how to leverage this potential. Therefore, we created a 2-stage digital health accelerator program to drive translation. We start off with a stage 1 accelerator leading to prototypes. We identify technologies and concepts, and then we do rapid prototype development. This takes about 4 to 5 years, and this is being funded by BIH. We mentor, we coach, and we give co-working space. The best prototypes then will be further developed to build certified, regulated, and validated products. So we do iterative product build, and then ideally at the end of approximately 12-month period, we try to spin off this project and make a company out of it in case we find a founder's team. So the preferred market approach is spin-off, but in case we don't find the right conditions, we do co-development with partners or we do tech sales. Everything to improve patient outcome and to get a very innovative solution to the market and help patients. We set goals a couple of years ago, 3-4 years ago, what we would like to achieve. And we identified that we would like to create 45 prototypes, about 24, 25 products, and do 15 spin-offs and about 5 license deals. And I would like to introduce, after a 4 years period, our first results we achieved. So we have a strong year-on-year growth in our program, meaning there is about 67% increase of applications. We have 45% increase in the number of projects being funded. This is the period between 2017 and 20. We reached the proof of concept where we showed that our end-to-end methodology, starting from a concept to basically getting the solution to the patient, was successful. Last year, within Corona, we were able to do 4 spin-offs. 3 out of them were funded with up to 5 million by venture capital, and 1 spin-off is a non-for-profit company in the developing world. In addition, we will move into a new location dedicated for co-working space to expand our activity at Charité and to build up an ecosystem in Berlin. We further will scale our digital labs activities to more research hospitals. So we'd like to expand what we have been building up at Charité to 10 other German university hospitals. And we already attracted 1 hospital, this is the University Hospital in Bonn, who would like to join this initiative. Lastly, we are currently in a fundraising process to complement the public funding we currently gratefully receive with private funds. Let me give you a short overview on what kind of spin-offs we realized last year. So the first one is called Agnostics. It's an AI software which identifies cancer cells and non-cancer tissue in histological samples. So it was coming from a pathologist. The second one, it's Dental XR. It's an AI-based diagnostic software tool to diagnose dental diseases. The third, X-Cardiac, a very nice and unique monitoring solution for earlier detection of complications in an ICU. For example, if you have internal bleeding or renal issues. The last one, it's a non-for-profit company, Mtomadi. They empower access to healthcare in developing countries by providing a mobile wallet, a mobile health wallet. So they do claim validation and quality insurance and connect patients, providers, and sponsors. Now that we have been successful working at Charité, our vision now is that we would like to develop the largest portfolio of translational digital health solutions originating from clinical research in Germany, all with the aim to improve patient outcome. The goal in this respect is to build and create about 110 to 120 ventures or spin-offs coming out of these research clinics. As I just said, there is about 15 to 20 coming out of Charité, and we aim for having 100 more from numerous different research clinics. In a nutshell, we have been successful in proving this methodology at Charité. We already attracted a second university clinic in Bonn to implement digital labs. And we are currently in an acquisition process to go for more research hospitals. The funding model is attractive because it is mainly originating from the states where the research hospital is located at, for example, North Rhine-Westphalia, or it could be Lower Saxony, depending on where the research clinic is. The BIH contribution is we deliver the blueprints, meaning the methodology. We deliver know-how and send experts to give a quick head start and to avoid basically mistakes we have been doing at the beginning. We hand over standard templates, and we are currently in negotiation whether we are able and whether we are willing to support with some finances. The refinancing model for BIH is to get some proceeds from possible exits, or we get some proceeds from tech sales, for example. If I summarize our initiative, we have been successfully building up this at Charité, and we are currently in a scaling process to give other research clinics in Germany the opportunity to use this methodology to foster translation. Thank you very much.
Thank you very much, Klaus Nitschke, for this very nice example and illustrating the insights of the model that you have generated. While people probably, if you want to have questions asked to Klaus Nitschke, put them in the feed and I'll read them. In the meantime, probably 1-2 questions from my side. A little bit a nasty question could be, well, those are very nice examples in the last year under COVID conditions. That was tricky for startups is fantastic. Yet 3 are diagnostic related. Is it easier to get, like in other fields of medicine, is it also here easier to get diagnostic strategies off the grounds than therapeutic ones? Or is it just accidentally that this happened? Or do you have a filter that you provoke this direction? Why do you see this?
We don't have a filter. So usually we take the applications which come from the researchers and we see trends. So the last 3-4 years, many diagnostic tools were developed based on AI. Now what we see is in the last cohort, there is mental health, there is telemedicine, and it changes a little bit going away from AI-based image analysis. Diagnostics is easier than therapeutics. I mean, if we will be able to do a digital therapeutics, this would be really great. But this is, let's say, the art of digital health. We are currently having a mental health solution to prevent gaming disorder. Currently, it is a detection tool, and we think at the same time whether we can further develop it to a digital therapeutics, but currently we still need to work on this model. To answer your question, a diagnostic tool is way easier to have a therapeutics based on digital technology or with the help of digital technology.
There is a direct question from Jochen Büchel that I will forward to you, and because it's a very direct question, who could collaborate with whom? I will send that further so that Jochen Büchel knows that we are taking care of the questions asked. What I would like to ask, sort of following what we just had, is the key measures of success. In the initial illustration from you, I saw that you had the idea to come up with 15 spin-offs and 5 licenses or licensing out. The ratio has a certain background. Maybe you have a word on that, why 15 spin-offs and only 5 licenses. But the question would be even further, isn't venture capital money not a better measure than numbers of spin-offs? So how much money others really invest in those structures as a risk capital? And why limiting to German hospitals, and why not going European-wide or even globally?
Yes. So first question with regards to ratio, spin-offs versus licenses. So what we realized over the years, it's quite hard to get a patent. Usually people do software. You can get a patent on a service, meaning software plus, let's say, some service being attached, or you can get a patent on some software in combination with hardware and service. So it's a solution. So in this respect, we prefer spin-offs because the IP is not as solid as you compared to as you have, let's say, a non-digital solution. And this is why the majority, at least this is what we currently plan, and this is what we see as well, that we focus on spin-offs or tech sales, and this is the basic thinking behind. And yes, you are right with regards to what's a KPI or a key performance indicator, or how to measure basically success. Absolutely. First, it's not just the number of spin-offs. You need to be sustainably develop them in the market, and you need to survive. Depending on, let's say, the impact in the market, you attract more venture capital money. So if we are able to attract a substantial amount of VC money over time for the spin-offs we created, this is a measure of success as well, because then this spin-off will reach more patients or more customers at the end of the day.
And a quick reply to Germany versus Europe
yes.
perspective.
I already approached the European University Hospital Alliance members with this let's say, methodology. And we are currently in talks and negotiations. Now it's a bad time because of Corona, because they really have other things on their plate. The current and the major bottleneck is funding. One needs to consider that what we do here is we need to have, let's say, small money to build a prototype and quite significant funding to build a certified and validated product. So it's not just, let's say, selling coffee, it's a serious product which needs to be developed. So if, let's say, if I go to Italy, to Milan, to San Raffaele, for example, they say, whoa, this is great, we would like to have it. So do you have some money and can help us to implement it? And this is true for Barcelona, and this is true for some other universities. And then there are some universities who are rather advanced with translation, so they don't have a need. So in this respect, I already introduced this to other research hospitals outside of Germany, but so far we are still in the process to let's say, get to an agreement. We haven't reached one yet.
Thank you very much. We are unfortunately running out of time. Thank you very much for the presentation. And a key question that just sort of Brenna asked, how do you identify and assemble the management talent to lead your spin-offs? I think this is one of the key questions to get those people together then in the end. I think you can think about those, a great answer for that. I think there is not really a very simple answer to find the right talents, but it's a challenge.
It is the challenge, and we have an extensive network within Berlin where we identify founders who are interested and capable. So far, we have been doing quite a good job. And now we reach a stage where people approach us and ask us, so listen, I already founded a company, I did an exit, I'm currently bored. Do you have anything which I can drive forward? So but still, it's a bottleneck, but we are doing quite good.
So if anybody has a need for finding a competent person, then approach Klaus Nitschke. He has the contacts. Thank you very much, Klaus Nitschke. And we're coming to the last part of this evening here in Berlin, evening event, midday event in the Boston East Coast area, and early morning for Utah and the West Coast, and very late for the Japanese and Asian areas, which we are very sorry about. What we would like to now do is come to the stories challenge at Translate, and we have now prepared the 3 pitches that we would like to share with you. Challenge at Translate, and we will invite you afterwards to vote which one you like most. After we had a chance also to talk to 3 representatives of the pitches, Albert Bravo, Christian Stephan, and Dimitrios Wagner, and the pitches should be shown now. Mats ab.
The whistle is a very simple and easy to use pocket device that is meant for a very quick test, 70 seconds, by a physician, not only cardiologist, in order to early detect heart failure. The whistle sends the information over BLE to the mobile phone. Here we measure the pressure that we built in our thorax, and this pressure is applied on the heart, and the heart is changing the pulse wave that we measure out of our fingertip using the camera and the flashlight. According to this change, we're able to say whether your heart is in order or not. Patients that suffer from chronic condition of heart failure experience very difficult day-to-day life. Basic activities like going up the stairs becoming a struggle. But now I think we found a way how we could prevent it from happening at all if we catch it on time. So it's a mission. So we are about to get our CE mark May 2020, and from there on we will be throwing clinical trials. So let's assume a patient comes into a physician's office and he's prescribed medication. But the way the physician finds out about the efficacy is subjective because the patient is just telling him about it. What we developed is an objective tool to see how the patient really responds to the medication, to the treatment, and it's a breakthrough.
Dear audience, thank you for your interest in my presentation. My name is Christian Stephan, and I'm a managing partner of the Kairos company. We are part of a research project so-called Sepsis DataNet, and it's a ministry-funded project. Today, 160 patients will die on sepsis, and the physician has no possibilities to predict who will survive and who will die within the next 30 days. Therefore, we established at 6 university sites our software solution as a local data management system and collect the intensive care data and as well the hospital information data within our system. After this, we transfer the data to a central data management system in a pseudonymized way and with the informed consent of the patient. Together with the sample information and the omics data coming from these samples, we build up an NI model to predict at least with 90% area under curve the 30-day survival of these sepsis patients. And therefore we build up a decision support system for physicians to make these predictions. Thank you for your interest and enjoy the other presentations.
Today, CAR T-cell immunotherapy is used as a potent treatment option for patients with refractory leukemia. When a patient is selected for this therapy, the physicians extract the patient T-cells and send them to a manufacturing site where viruses are used to genetically introduce chimeric antigen receptors randomly into the cells. These CAR T-cells are then sent back to the physician who can use them to treat this and only this one patient. Oh, and this entire process takes about 1 month and comes with a 320,000 euro price tag. That is why our team here at Charité is working on the CAR T technology of tomorrow. We have successfully developed a new platform technology that enables us to produce CAR T cells using genome editing and synthetic materials only, eliminating the need for viruses. We can create multiple doses of CAR T cells from a single healthy person ahead of time, so that when the patient comes in, physicians can immediately treat them, but at a significantly lower cost per dose. We have established this procedure in the lab, and we are now ready to scale it up for clinical use with our GMP experts. If you want to make CAR therapeutics more affordable, want to learn more about our technology, or create a new CAR therapy for a disease which you are specialized on, please contact us.
Yeah, thank you very much, all three. for your pitches and for your contribution. We are very eager to learn a little bit more about how you came that far and what you eventually envision for the next challenges and the next steps in front of you. Probably just to get it started, what was the biggest challenge you experienced on the way to be where you are at the moment? What would you mention was the biggest challenge to overcome? And maybe we simply, if Albert could start, for example, and then Christian, and then Dimitri. You're muted. I don't hear you.
Oh, sorry.
Yeah, better.
Thank you for inviting us to this amazing event. It's an honor to be here. So we experienced very similar problems as Mr. Klaus Nischke has just introduced. We face issues with patents exactly as he said, but the most troublesome topics that we have encountered is the regulation. And unfortunately, it's becoming even more severe this year. The directives of the CE mark are switching from MDD to MDR, and CE mark will become more or less like the FDA. It's making life difficult, even more complicated as it was before for little guys like us. I guess that's why what Charité is doing together with Mr. Klaus Nischke to go for the quantity that might turn into quality is a great strategy, I guess.
Christian, would you comment on what was the biggest challenge for you so far?
Yeah, thank you. Thank you for the introduction and to present our project. So the most problem in the project was to get the people involved, and the people are totally distributed. So what does it mean? The people coming from the research part, there are people who are coming from the IT tech part, and we have so many information who should be come up at least from the researchers, for the physicians and the patient itself. And that's a problem because you have to integrate all these data. And these data is totally distributed in a normal university hospital setting over several different systems. So the dialect they are speaking in every and each university hospital is totally different. So therefore we need this data integration. We need this informed consent of people. We need this agreement about any kind of centralized description. We call this a common data set, where they all agreed to send their data in the same format. And that's very difficult because this data, which is integrated locally in one system, have the same meaning of the central definition, but has other words. And therefore we need this ETL process, these interoperability to send these data at least in one central database. And then that's the starting point where it becomes easy. Then we can start to build up AI models. Then we have this possibility to use statistical methods to come up with any result. But the first part is even bigger and even more difficult than the analysis itself of the data.
Demetrius, from your perspective, what was the biggest challenge so far?
Thank you. Yeah, I think we are the project that is earliest in their phase of translation, and I think for us the hardest part was actually to getting the science right, and maybe pointing to some of the questions that were asked earlier regarding reproducibility. We first went into what other people did and how other people use these kinds of technologies that we are performing right now, and we actually had to re-optimize a lot of things because some things were not as reproducible as they appear to be, and that's why getting the technology platform up and running was hard work and a lot of frustration in the beginning, and being able to get this frustration tolerance is, I think, very important at these earlier steps.
Probably to all three of you, a brief question. Would you, in terms of what is the biggest challenge that you see in front of you? Is that the knowledge of the regulatory part that Albert mentioned, which MDR regulations might be different, might be also not so completely clear how they will look like for that specific device, or how the regulatory authorities will eventually rank that? Is that probably the same also for sort of going deeper into the regulatory framework from the three different perspectives that you touch? Is that the biggest challenge that you still see, or do you feel you're already across that valley? Or is the venture capital question the biggest that you experience, or that you are personally feeling as the biggest next challenge? Who would like to?
You're asking Zenco Labs.
Yeah, for example, where do you feel yourself?
Our eternal goal, as was mentioned before, pretty much is an internal goal of any diagnostical startup, is to turn these diagnostics into therapy eventually, to hook it with a therapy and create a closed feedback loop through medication and diagnostic, or medication-less therapy, but based on the diagnostic that we create. And it's two different phases of product life. Also in terms of regulation, because regulation for a therapeutic product, for example, that is built on top of a diagnostic product, will change and become more severe. So it is indeed a plan that has to happen beforehand, before the execution even of the diagnostic part. And the company must plan ahead how to create the diagnostic solutions so it would be able to become therapeutic eventually and generate much more usability, become simply more useful, because eventually what saves human life is therapeutics, diagnostics as well, but therapeutics has much more effect on it. So, yeah.
Thank you. Christian, from your perspective, what is the biggest challenge that you think is in front of you?
You're getting totally right. So the problem is today, the data protection and the data security issues, and as well, it's very important to have a good quality data set, a good quality product, that you have to have your software solution as well as a medical device. And you have to understand this, that everything you are doing and any kind of decision support you support as an expert system to a physician is one of these medical devices. So the regulatory are very high, but they're very important to get up at least with high quality decisions, I would like to tell at the end. But in the starting point, you have to overcome all these hurdles. You have to talk to so many people. You have so many paperwork for these regulatory as a informed consent, starting with data protection rules and all these things. But later on, it helps you to have it even easier to collect these data, and also to get in touch with the patient maybe later on after they leave these kind. And some of these, and we know this for COVID as well, diseases are not only one-time diseases, so they have several problems after leaving the clinic, and therefore it's more important to understand what was prior these data we're getting and what will be after they leave the clinic happen to these patients. And all these together has been done, and this is one other issue where we have to tackle with.
Thank you very much. Last question goes to Dimitrios. What do you see as the biggest challenge in front of you, getting that into reality as a company?
Well, first, it would be nice to first have a small-scale clinical study, and towards that end, I see two big problems. Every time you use a new technology, there are no clear paths ahead because it is not established yet. So there are multiple problems that I think we will face when we prepare this for a clinical trial, which is first, obviously, the regulators don't know what is the best way to assess this technology. And then the second point is also supply chain, because it is a new technology. We probably have to establish some of the supply chain ourselves. For example, where to get the synthetic DNA, who's the best partner for that, and who can provide it at GMP grade. I think these are two very important topics that face ahead. And then let's see for the turnout for the first clinical trial, and then afterwards we can think about how to best market this and how to position us in this highly let's say full field with a lot of competitors.
Thank you very much, all three of you. I invite now everybody else who listened to vote and pick the best pitch. I think they're all three fantastic, and we're very much looking forward to what the auditorium is in the end then choosing and picking. Thank you very much for your time. It's the end of the first day. I enjoyed very much the discussions and joined together with you the meeting, and I hope you got inspirations, ideas, and appreciated the discussions that we had. Please make also use of contributing to the consensus papers with your thoughts and criticism. We're very open for those ideas. Vote for your favorite finalist and do that as fast as possible. We're going to announce the winner at the end of the second day. You already realized how much wrap-up we have heard for various aspects. Regulatory topics will be tomorrow a big topic again, and we'll move further. We'll have a startup tomorrow morning with US time, not tomorrow morning, here in the afternoon, with a wrap-up of today's day by Dave Granger, who did an enormous work in preparing this meeting jointly with all others, and I'm very much looking forward to that and his introduction of his keynote speaker. And I thank you very much for your attention, and with that, I'd like to end the day, and thank you very much for being with us.